Okay, why don't we get started then? So I'm Steve Carr. I head up the proteomics group here at the BroadE Institute. I'll be giving the initial overview of proteomics as we apply it here at the BroadE. And that'll be followed by each of the groups here discussing immunopeptidomics, applications in biology and translational medicine, single-cell and spatial proteomics, hydrogen deuterium exchange mass spectrometry for protein dynamics and interactions, affinity proteomics, proximity labeling and target identification of small molecules. And then that will end with a really important section on data analysis. How do we help you interpret this data? How do we interpret the data? What are the new cloud-based tools that we're developing? Okay. So I'm going to begin with kind of a proteomics 101, not going into enormous amounts of technical detail, but what I think is fairly important for you to understand. So these are pictures of 4 different mass spectrometers here on the screen. They all do a number of things in common. So I don't know if this is working or not, maybe not. I'll use the pointer here. So they create ions from analyte molecules. So we start out with things in the liquid phase and convert them into the gas phase and then cause analytes in that solution to ionize. We then separate the ions that are produced based on not just their mass, but their charge to mass. Okay, it's very important. And we detect ions and determine their mass to charge. And for actual structural elucidation, we then select and fragment ions of interest. That gives us the sequence if it's a peptide, for example, or a protein, or partial structural information if it's small molecules. In modern proteomics, really doesn't matter how you approach this, these steps are pretty much in common. So you have biological samples, which can be case control, mutation versus wild type, drug treated versus no drug treated, knockouts, all those variations. From those samples, which can be cell lines, can be mouse models, clinical samples, and we're going to show you examples in each of these areas during the course of the presentation today. Biofluids as well, like plasma. We take those complex protein mixtures and we digest them to peptides. And usually we're using an enzyme like trypsin, which cuts after arginine and lysine. This is kind of a non-intuitive thing to be doing because we take an already horrendously complex mixture of proteins and make it, 100 times more complex. But the reason we do this has to do with the data analysis piece at the end here, which has been fine-tuned and honed for the analysis of tryptic peptides. So we can't just shoot that entire mixture. Well, you can into the mass spectrometer. The problem is that we're limited by the dynamic range of what the mass spectrometer is able to actually detect. So it's going to be limited by the most abundant things that are present. For example, if you're in plasma, it's going to be hemoglobin. And that then limits, because you only have about a 10 to the 4 dynamic range, the bottom, the most sensitive thing, the least abundant thing you're going to be able to detect if that's the process you use. So we don't use that process. We fractionate the samples at the peptide level. Then each of the fractions that we generate is far less complex than the entire mixture. Each of those fractions is then analyzed on the mass spectrometer. And then at the end, when we do the analysis, we put all that data back together as if we had run it as a single sample. This way, we're able to get much deeper into the analysis of that particular complex material. Importantly, if we're interested in post-translational modifications, and we almost always are interested in post-translational modifications, it's certainly one of the things that sets mass spectrometry-based proteomics apart from genomic methodologies. Generally, we don't do that by direct analysis of the proteome and hope we're going to be able to see sites of modification on proteins. That's doesn't work all that well. So what we use are enrichment strategies, and we'll come back onto those during the course of the presentations today. Usually they are antibody-based enrichments at the peptide level, or can be metal affinity enrichment, which is what's commonly used for phosphopeptides. So that mixture is then separated and analyzed at the peptide level by LC-MS-MS. That's liquid chromatography tandem mass spectrometry. And I'll talk about what that tandem piece is in a moment. From the mass to charge and intensity of the peptides that we observe, this is a rich pattern. It's not identification. It's abundance pattern that once we identify, we can go back to that precursor peptide and say, okay, I can use the intensity of that peptide to say in sample A versus sample B and come up with some quantitative information. I'll come back to this in a moment. But we generally then fragment the ions for sequencing. Okay, and that's done under instrument control. And this is a process called data-dependent mass spectrometry. We fragment ions also in order to localize the site of post-translational modification. All of this fragment information, which has not been analyzed at this point, goes into a data analysis program. At the BroadE here, we typically are using a program called SpectrumMill, which was developed by Carl Clauser. And we have full control over that program so that when modifications to it are needed, Carl, with the help of others, goes in and makes those changes so that new sample types or extremely large sets of samples, we now have the ability to analyze those using that particular tool. What we're searching here is the fragment information and the precursor mass of the peptide against a database. I call this looking up the answer in the back of the book, because fundamentally the book is the translated genomic information that we are accessing. So we take the genome of the organism we're interested in, we reverse translate that to protein, and then we cut up that protein in silico to produce fragments that we would expect to observe. We'll come back to that again in a moment. For post-translational modifications, that the database, if we tell the system, go look, make sure you look for phosphorylation, look for ubiquitylation, et cetera, it will actually extract that information from the data that we're providing as well. Out of this comes protein identity and the relative abundance based on the intensities that we're seeing of the peptides, and that's rolled up to the protein level. So this is one way of getting quantitative information. So starting with the basics here of how do we sequence peptides using data-dependent acquisition. So at the top of the figure here, I'm showing you just the initial mass spectrometry step. And I'm showing this in the simplest way with 3 regions. The first mass spectrometer that is scanning over a mass range that's decided by the user of the instrument. So here I'm showing mass range 1,000, mass to charge 100 to 2,000. There's a collision cell here, which during the MS only section of the analysis is turned off, okay? We're not doing any collisions in this cell. So it's passing all of the ions that are being transmitted during the scan of MS1. Pass all ions, and you produce this spectrum shown here, where each of these ions represents either some charge form of a peptide. It could be singly charged, doubly charged, or triply charged. All forms can be observed. The charge state that you see depends on the readily chargeable residues in the peptide. So typically, if you have an amino terminus of a peptide, that's free, and a lysine or arginine in the peptide, that produces a doubly charged peptide. If you also have a histidine in there, you will then get a triply charged as well as a doubly charged. So some of these rules are well worked out. You select an ion, and this is done under instrument control, not user control. We are not sitting there going, oh, select that ion and fragment it. The instrument is looking at the spectrum that's been produced and says, I'm going to start with the most intense ion in that spectrum, fragment it, then move on to the next most intense ion, et cetera. And it might do this over the top 20 ions in that spectrum. It'll then stop, and a new scan will occur in which it now says, I'm not stupid. I'm not going to reselect the ions that I already selected. I'm going to move on to a new set of these. So this is the way the instrument then digs down deeper into each of these spectra. The fragmentation of this ion right here, this doubly charged ion at mass 836.5, produces this fragment spectra. They don't come out labeled like this. Wish that they did, but the software, like SpectraMill, then interprets this and can produce a labeled spectrum where things are being fragmented either with charge being retained on the N-terminal portion of the peptide or on the C-terminal portion of the peptide. And we call these B and Y ions respectively. That level of detail, I think, is beyond this right now. I have a whole series of papers that I list at the end of the presentation. You'll have access to all of these slides and the references after the day. What do we do next? Well, putting this all together, this is the full kind of workflow. Start off with samples, extract proteins, digest the peptides. Those get shot onto, maybe with pre-fractionation, as I talked about, onto the LC mass spectrometer system. This is using acidic reverse phase. Offline, we might do basic reverse phase in order to get further decomplexing of the sample. Then each of those, as the mass spectrometer is scanned, it produces what we call a total ion current trace. So this is the equivalent of a UV trace, if you will, if you ran a complex mixture on a HPLC system and monitored it by some UVs. Except here, it's total ion current. These are representing peptides as they are eluting from the mass spectrometer. The next thing you do then, as I mentioned already, is that they are selected by the instrument on the fly, usually 1 to 2 seconds per scan duration. It selects ions in the mass to charge window of interest, fragments each of those, and then the instrument picks, as I said, top 10 to 20, sequences all of those. That's what this card is showing, multiple fragmentations. And all of that at the end of a run is fed into the database search. Again, just to reiterate the process here. So we have acquired MSMS spectra. We have reverse translated database, translation of the transcriptome to produce a model spectrum. Here I'm showing that there's no intensity information, only the fragment masses. Nowadays, software has gotten sophisticated enough that it is capable of also predicting what the relative intensities of these fragment ions would be for any given sequence. It's getting very good. At the end of the day, what you're doing is correlating, producing a similarity score between the observed spectrum and the model spectrum. And we end up saying this peptide is what we're looking at based on best match in the database or spectral library. On top of that, we're searching, as I mentioned, for post-translational modifications, as well as sample handling artifacts. So, for example, you always get oxidation in methionine. That's taken into account when we do the search. We also look for common contaminants like keratin, and we basically say, yeah, they're there, but we set those out of the picture. This process can miss unanticipated modifications. So if you weren't expecting arginine methylation, for example, and you didn't purposely search for it, you may overlook it. Likelihood is you would. So you have to be smart enough to go looking for things that you think you're interested in. There is a way of determining the false discovery rate for these peptides at the simplest level. As said, we're doing a forward search against the in silico digest of the database, human database, mouse, whatever it happens to be. And for false discovery rate determination, we will we'll hold the two end residues of the tryptic peptide constant, but reverse all of the amino acids in between in their sequence. And then we ask the question, how many of those matches do we get? Okay, that gives us a false discovery rate for the peptide. And then beyond that, we produce a false discovery rate for protein level. There is a future here, de novo sequencing. We're not quite there yet. Okay, that is still in process of being perfected. What do I mean by de novo? It means you don't do a search against a database. You actually directly interpret that spectrum through machine learning approaches of one type or another. I've been in this business long enough that I am the AI 30 years ago. I would get a spectrum and I'd be asked, what is the sequence of this thing? And then actually on paper work it out. When we have new people in the group, we actually have a course on making people actually interpret spectra, because we want the students in the lab to understand how the sausage is actually made. Okay, and then we get lots of peptides that are associated with any given protein. So here, peptide 1, peptide 4, peptide 7, all are associated with protein A. So probably it's protein A that is what these peptides are associated with. And similarly, we have cases where there's only a single peptide that is pointing to a protein of interest. We generally, and it's only generally, we like to have at least 2 distinct peptides, ones that have different sequences from any given protein that uniquely associate with that protein before we'll call that protein. Nowadays, those rules are getting a little bit looser because the quality of the data is getting so much better that the sequence information, even from a single peptide, is often sufficient for us to be confident that we can identify that, particularly when we continually identify that peptide over many samples. So now we have lots of peptides that we're observing, some of which can associate with protein X, others associate with protein Y. And then there are peptides like this that could associate with either protein X or protein Y. And then the question is, what do you do in a case like that? There are options. You can assign that peptide to the protein that has the most peptides associated with it. This is what we call parsimony, or Occam's razor principle. The other way to do this is to assign it to both. Okay, you just need to be clear as what you're doing and understand why you're doing it and what the implications are if you do that. The other is to ignore shared peptides. We don't think that's a good idea, honestly. The other is to assign it to the protein with the most similar quantification. That can be dangerous if you don't have really strong signal for that peptide. There may be a lot of noise and you can make a mistake when you make that assignment. So there's a lot of criteria that are being applied when we make these sorts of judgment calls. So at the end of the day, we end up with proteins that we've assigned in or samples in state A, state B, drug versus no drug, bait versus control, et cetera. You understand the concept. And just like you do in transcript profiling or most other approaches, you then come up with some kind of a scatter plot or a volcano plot where you're looking for things that are being regulated and you're modeling this based on t-statistics or Gaussian mixture modeling. You can apply linear modeling. You'll hear more about these approaches from Mani and Natalie at the end of the day. But generally, we run these things in replicate. So this is replicate 1 on this axis of drug versus no drug, replicate 2, drug versus no drug, and we get a nice line on the axis where what's in the upper right corner here, based on the way we've designed the experiment, are things that are specifically upregulated in the presence of the drug. Things down here are potentially downregulated in the presence of the drug. Things that are off axis tend to be noise in the system. So now I'm going to tell you 3 ways that we approach quantification in proteomics. The first of these, and the most in principle, most straightforward, although it's not, is label-free quantification. This is where you have 2 samples in state A and state B. You extract proteins, you digest proteins to peptides, and then you run those samples individually and then do a comparison of the peptide 1 in sample 1 to peptide 1 in sample 2. So you're doing a 1-to-1 correlation across these 2 different samples. And that's what I'm showing here as a ratio at the precursor level, the intact peptide level. This tends to be less precise as you go to lower and lower intensity of the peptides, but there's now a new approach, and I'll come back to it, called data-independent analysis, where this is really the way you run the samples. And we'll talk about that in some detail. Second method, which used to be very commonly used in our lab here at the BroadE, less so nowadays, is metabolic labeling with labeled amino acids in cell culture. So here you have state A and state B. These get fed, they are fed one isotopic form of the amino acids lysine and arginine. And state B, where they're fed a heavy isotopically labeled version of those amino acids. What happens is that after a certain number of cell doublings, all proteins in these samples incorporate either the light form of those amino acids or the heavy form of those amino acids in all of the proteins that are being made. We then can combine the samples together, shown here, and digest. Now, how do we sort this out? Well, any peptide that has incorporated heavy amino acids will be shifted by the number of carbon-13 or nitrogen-15 in the heavy version of that amino acid in each of these peptides, whereas the light version will look like the wild type. So and you'll know that the heavy ones came from sample B and the light ones came from sample A, and therefore you can ratio, again, at the intact peptide level to come up with the quantification for that particular peptide in that sample. And just like all the other measurements, we then roll up peptide quantification information to understand what the protein quantification is in that case. This has somewhat limited plex level. People have done this up to 3 different states, sometimes even more through some tricks, but it's limited. And right now humans can't be labeled. You can label mice. It's extremely expensive because you have to feed them these heavy labeled amino acids. So the approach that is now typically used for these is not isotopic labeling in cell culture, but chemical labeling. And for this, we're talking about things called tandem mass tags in particular, or iTrack reagents. The most versatile of these, because it has the higher plex level, are the TMT reagents from Thermo. And here, while I'm still showing only 2 states for simplicity, you can now have up to 18 samples in the multiplex. Each one gets a different chemical label that is distinguishable after you fragment the sample. Now, I'm not going to go into the details of how this works, but just realize that here the identification is based on the fragmentation of the peptide as before. Now we're getting our quantification at the MS level, not the precursor level. At the precursor level, these things are isobaric. That's the trick. And you can look, I have some papers that you can read up on this. I just think it's too much for this morning. When they fragment, when the peptide fragments, it releases the tags that are on these peptides, and those tell you from which sample that peptide has different ratios. And again, as I mentioned, you can do up to 18. You don't have to do 18. You can do 6, you can do 10, you can do up to 18. And one of the problems here is that there is some issues with compression of the quantitative information. So what I tell people generally is if you see a difference, if we see differences when we run the TMT experiments between state A, state B, whatever it is, it's real, you can believe it. However, if we don't see a difference and the signal level is low, can't be absolutely sure that that particular protein is not changing in abundance. To get better quantification in these samples, we fractionate very extensively, and you'll hear about that in multiple talks. And the reason for doing that is by decomplexing the sample, the quantitative, this compression business is reduced significantly. Big problem with these is that the reagents are expensive. Okay, and I mentioned post-translational modifications. Really, there's a zoo of these things. The 3 that I've listed here are among the most common and of biological interest. today. I also list arginine methylation. We can do that. Oligocalation. We and others have developed methods for doing this, and sumylation is something that is on our radar. There are some methods that have been developed. They're not perfect yet, but we think they can be made better. And of course, there's lots of other palmitylation, tons of different modifications that we can, if you're interested, and approach us, and there's a good project, we can talk about developing that modification, a way to analyze those modifications. So at the end of this, we have the acquired data that we've analyzed, and gets put into data tables using SpectroMill. So all this raw data after analysis from the mass spectrometer coming out of SpectroMill produces information showing protein 1, protein 2, protein 3 across the different samples here. So you get out information about the peptide in the protein and the abundance ratio across the samples. I want to emphasize ratio here because the information that we provide is not absolute. We do different experiments if there's a need for absolute quantification. This is all relative quantification. Sample A versus sample B for that protein, that modification, et cetera. You'll hear much more about this later in the talk today. These are tools that this group and others have developed, but Prodigy is home built. This is a tool set that takes the data tables of proteins and quantitative information coming out of SpectroMill and other tools and allows users, not just the BroadE proteomics team, but those interested from our biological collaborators to go in and do new normalizations, new types of statistical analysis, point to different spots on the volcano chart and ask what protein is that. It's a very robust and useful set of tools constantly being worked on and improved. We have tools that have been specifically developed for targeted mass spectrometry. We'll go into detail about that today. I talked about SpectroMill already. You'll hear about Panoply from Mani and from Natalie later today. This is a suite of tools called cloud-based that is meant for as automated and reproducible analysis of proteogenomic data, where we're now taking the genomic data that's been generated for patient samples, overlaying the proteomic information for exactly the same patient samples and all of their modifications, integrating that information, and then producing analysis results, graphics ready for publication, et cetera. And this is a tool that's really under rapid development, and it's kind of open source to some extent, and you'll hear more about this from Mani and Natalie. I'm going to skip this. This is just general things that these tools do: clustering, correlation, marker selection. So I'll let them talk about that. These are project areas that we engage in within the group. I'm going to touch on just a couple of these today. You'll hear from Jenn Abelin about our immuno-oncology efforts and immunopeptidomics, drug mechanism of action. You'll hear about from Namrata, signaling and pathway information in proteogenomics from Shanka. You'll hear about structural proteomics applications from Malvina. Yes, and then you'll hear from Mani and Natalie about tools that we're using both within the group, taking from the community, and then enabling our community with them. Okay, jumping into just a few applications, highlights, because we're going to go back and touch on these in the individual presentations. So this is a kind of just a highlights reel for the next several slides. So mentioned multiomics and proteogenomics. We and others are part of this National Cancer Institute Tumor Analysis Consortium. I've been in this now for 16 years and still going. These are some of the tumor types that have been analyzed. My group has done lung adenocarcinoma and lung squamous cell carcinoma and breast cancer in the past. Currently, we're working on sarcoma and thyroid. And in these experiments, we're getting from prospectively collected materials, tumor samples curated by the National Cancer Institute, generally about 100 or more of tumor samples and adjacent normal if available. Here are some of the publications. And again, you'll have access to all of this in the slides. In order to do these sorts of analyses, we have developed robust multi-omic approach for analysis of tumor cell material. And recently, you can read about it in this publication, Jenn Abelin and the team at the BroadE developed methodology that now can even start from immunopeptidomic data. So we can generate from a tumor sample very rich and deep HLA class 1, HLA class 2, and then take the flow through from that sample and continue on to do ubiquitylation, full proteome analysis, phosphoproteome analysis, and acetylome analysis. And in principle, you could take flow throughs from this and continue on. We also do phosphotyrosine specifically as part of this. So the advantage here is that from a single patient sample, we are extracting all of this information. We're not asking for separate sample aliquots to do this. So it's very, it's sparing of precious tumor material and providing a lot of information. Shanka, in the group who you'll be hearing from later, we worked with collaborators at Baylor College of Medicine to develop a microscaled approach where we're able to take core needle biopsy samples and from a core get DNA and protein, RNA, and even non-denatured protein from that core for affinity-based assays if necessary. Here, because the amounts of material are limited, we get fewer proteins, still very rich proteins, 11,000 proteins per core. Phosphocytes, 20,000. Well, it's kind of a meaningless number for you. You give us more material, we'll give you 65,000 phosphocytes these days, 20,000 ubiquital sites. It's very, very deep coverage of these various omes. There's a paper on this that you'll see. From Namrata, you'll be hearing about both target identification, as I mentioned, but also this fascinating area called proximity labeling that uses enzymes, often peroxidases, to leverage a small molecule, in this case, a biotin or biotin conjugate, that upon reaction inside the cell where you've placed this peroxidase, you get a local reaction, proteins that are near the vicinity of this particular apex or other modified protein, and you then are able to say what proteins are in vicinity of that label. This is not direct identification of things that are bound to that protein. They're in the neighborhood. The neighborhood meaning 10 to 20 nanometers from where this bait protein ends up. So here I'm showing you 3 different versions. You'll hear a lot more from Namrata about this, but Apex is and Turbo ID are flavors of this that our lab has worked on with Alice Ting, who used to be across the street at MIT. She's now at Stanford, and we've been doing this with her now for, I think, 12 or 15 years. So long-term collaborations in this area. Importantly, beyond just doing compartment identifications, you can even do surface labeling experiments. So if you're interested in the surfaceome of a particular cell, you can now use either horseradish peroxidase bound to a surface protein or Apex for intracellular, and use a labeled biotin version that is membrane impermeable, okay, importantly. And then you only get labeling of proteins that are on the cell surface. There have been extensions of this work using for light temporal control, this Love Turbo, this just recently published, and for tracking proteins that migrate within an organism. You produce a protein, for example, in the brain, and you ask where that protein goes after it's been secreted from the brain. There's a transit ID approach that's been used for this. And again, you can look this up. So you can label in one compartment and then use a different label in other compartments and ask the question, is it doubly labeled? And if it's doubly labeled, it came from the source and ended up where you did the analysis. Affinity proteomics, again, you'll hear a lot more from Namrata here. So I think I'll skip this slide other than to say we use this for protein-protein interaction, but also protein DNA, protein RNA, and small molecule protein interactions. So all revolving around the use of a bait of some sort. Generally, in this case for protein-protein, it's a labeled protein. You either have an antibody to that protein that you're using for affinity enrichment, or you tag a protein of interest in a cell, in the context you're interested in, and then use an anti-bait antibody for the enrichment. There's a whole series of sort of QCs that we insist on that you do. So when you do these experiments and approach us, we give you an SOP for things you have to do before we'll accept any examples. Okay, it's all for your benefit. It's to make sure that we succeed in those analyses the first time around. Target identification, again, you're going to hear about this from Namrata at more extent, but how do you determine what a compound, particularly something you've developed in the course of a phenotypic assay, what it binds to inside the cell? All you know is that it does something. Generally, it kills the cell, but you have no clue what the actual target is. So in the past, the way we did this was to take the compound that was doing something of interest. We use the SAR information that we have for that compound and then modify it in a part of the molecule where we thought would not destroy the bioactivity. Then you'd have to follow that up with actually making the modification being putting a tether on, because at the end of the day, what we want to do is tether that small molecule to a bead and use it as an enrichment device. But you have to go through various testing to make sure you haven't screwed up the activity of the compound. It's time consuming, failure rate's non-zero, but we have done it, and we've done it multiple times. Problem is, it needs chemistry support, and that often is very limiting. So what would be transformative? Transformative would be ways where you don't have to actually tether that compound. And you may have heard about thermal proteome, certainly heard about CETSA. CETSA is this thermal shift assay. It's done commonly. It's generally one protein at a time that one does this for, and you're looking for changes in the solubility, basically, of a protein over the course of a temperature range in the presence or absence of a drug, okay? And the shift that occurs in the presence of a drug is telling you that protein is being affected. You can make CETSA work in an entire proteome-wide way. This is called thermal proteome profiling. And again, you're running a temperature gradient of a compound now stressing the entire cellular proteome, asking the question, which proteins are changing in solubility across a temperature gradient? And that's plus or minus the drug. The ones that have differential response in the presence of that drug are targets, potentially targets of that drug. That also is extremely complicated methodology, and there's a new version of this that Namrata will walk you through, so I'm not going to dwell on this slide other than to say that is proteome integral solubility alteration, has very specific advantages, greatly simplifies the whole process, reduces the amount of cellular material and drug that you need to use. It's not perfect. You no longer have the curve that you get out of thermal proteome profiling, but it is useful for telling you in a screening fashion what the targets are of your compound. And you'll hear more about that from Namrata. From Malvina, you're going to hear in detail about hydrogen deuterium exchange for protein interactions and dynamics. So for sake of time, I'm going to skip this slide. Now, in the last just few minutes, I want to tell you about a slightly different way of acquiring the data, which we're now making hay with in our lab. This is data independent acquisition, but I'm going to start by just telling you again what data dependent is. So this is data dependent acquisition. The instrument is going through and on its own selecting regions of a mass spectrum and then fragmenting it to produce fragment spectra that then get searched against a database. And it does this sequentially during the entire run. Problems are listed here. The sample is dynamic during that analysis. Only 20 to 50% of the peptides that are present are actually measured and fragmented, leading to a sequence identification. A lot of information left on the table. Increasing depth and completeness requires offline fractionation, as I talked about. Stochastic measurements result in missing data. So you may see it in sample, in particular, this is very true for post-translational modifications, where for proteins, you could see different peptides from that protein across different samples and still be able to identify that protein. For PTMs, you have to see that phosphosite repeatedly across multiple samples. And the dropout in your ability to detect that when you're at sample 100 versus sample 5 is really quite great. So there's missing data. The question is, how do you deal with that? And as in all of these cases, signal not detected means either the feature is not actually present or it's just below your detection limits. That's DDA. DIA is shown here. In this process, what we're doing is instead of telling the instrument to go off and select precursor peptide masses and individually fragment them, it's using a process where it basically just jumps M over Z ranges across the mass range you select. So across an entire M over Z range, for example, in 20 M over Z chunks, it takes a 20 M over Z window, fragments everything that's in that 20 M over Z window, moves to the next 20 M over Z window, does the same thing until it completes the mass to charge range that you told the instrument. And then it cycles back and repeats that process. So fundamentally, you are getting MSMS spectra for everything in the sample. That's great. The real question is, can you interpret it? So all peptides present are in fact fragmented. And the other advantage of this is that really high reproducibility. Those things are being repeatedly scanned and fragmented across your experiments. It generates overlapping fragment spectra. So you've got 100 peptides in a given window. The spectra that you get is the overlapping spectra of all 100. Complex. Data is generated in single-shot LC-MSMS. What I'm talking about here is we don't do any offline fractionation. We might run a 2-hour gradient. Sometimes we run a 5-minute gradient, okay, and collect this DIA data. So it's much faster. There's far better data completeness. Depth of coverage is generally, when done right, 70 to 80% of what we get when we do a DDA experiment with fractionation. At the proteome level, we're talking. And again, signal not detected means it's either not there or below detection limits. So it's just some examples of this that you'll hear a little bit more about from Shanka. This is DIAMS. This was something that we did with the DepMap team where we were profiling 55 cell lines, asking the question, what do we get when we use DIA in this very rapid 1 hour per sample analysis on the instrument? And what you get is on the order of almost 9,000 proteins detected in these samples, of which more than 5,000 are consistently detected across all 55 cell lines. Some of these are not there because they're not actually in that cell line, okay? So this is showing much faster data acquisition at the proteome level. DIA is useful, and the quantification information is actually quite solid as well, and you'll hear more about that. This is being extended now to formalin fixed paraffin embedded tissue, and from which we can now obtain deep scale proteomic information. And I don't know, Shanka, are you going to go into some detail about this? Yeah, so again, I'll skip this. He's going to tell you more about how we do this, but just want to make you aware, FFPE is wonderful because there are decades of samples stored in freezers with really extensive clinical information, which is extraordinarily valuable, that we can now tap and get information from that is valuable from a clinical perspective. Again, you'll hear more about sort of spatial proteomics applications where you can do scrolls from these FFPE materials, isolate regions, analyze them by laser capture cut them out by laser capture microdissection, and then analyze by mass spectrometry. And again, because you're going to hear about this from Claudia and Shanka, I'll move on. You'll hear about single-cell proteomics, which Claudia is our in-house champion developer of. She'll tell you where we currently are in this, and that's an old slide because that depth has been greatly exceeded at this point. Yeah, I'll skip this one too. So yeah, plasma proteomics is also very doable by mass spectrometry. And what I'm trying to show you is that we previously developed an extremely deep approach, getting very deep in the plasma proteome. The problem with this methodology is that it takes, it's sort of on the geological timescale because you have to do so much fractionation and so much analysis of even every individual sample. It just takes too long to be clinically valuable. Newer approaches have appeared where you can do kind of single shot analysis of plasma. This gets you shallow information into the plasma proteome, may be useful in some cases for metabolic diseases, but for things like cancer, unlikely to be of much use. Then there are other techniques that we have in the lab here that is using nanoparticle-based enrichment. So these are chemically derivatized nanoparticles that are used to enrich proteins out of plasma. They're then removed, the proteins are then isolated from the nanoparticles, digested and analyzed. And the main thing to remember about this approach is it doesn't just analyze the most abundant proteins. In fact, they're often some of the least abundant after you do the enrichment. So these particles are enriching things across a very wide dynamic range and not entirely dependent on what's at the highest abundance level. That's the SEER technology. We're applying this in the case of multiple myeloma. I don't have time. I'm running out of time. I should stop now in terms of the kind of information that we're generating here. But just to say that in our multiple myeloma studies with Irene Gobriel over at the Farber, we are able to replicate some of the known markers and finding things which are entirely novel as well. I'm going to just end by saying there are targeted MS-based approaches that differ from these discovery approaches, whether they be DDA or DIA. This is where I did my discovery experiment. I have a list of 10, several hundred proteins that I'm really interested in, and I now want to monitor across many, many samples. In the past, the way we would do this we would create, use heavy isotopically labeled versions of peptides from the protein of interest, spike those into our digested sample, and then ratio the heavy to the light form of that peptide. And this gives us an absolute abundance of that peptide, enabling us in the course of an hour to measure things, up to several hundred different proteins. Nowadays, the DIA methods are beginning to slowly eat into this capability, and we're sort of on the fence as to how much of this targeted approach that I'm showing you here versus an approach on using data-independent analysis might be able to solve. So just know that these targeted assays are available. We have a 300-plex assay we developed for phosphocytes called SIGPath, and we're ready to apply that, and we are applying it in a number of different cases. Right, this is another example in the case of multiple myeloma. Okay, to end, just hope that's a very rapid global view of the types of things that we do in the group. We are both a center of collaboration, but also a technology innovation and development center. So when you come to us to talk, don't come to us thinking this is the limit of what proteomics can do. Come to us telling us what is the problem you're trying to solve, and then we'll figure out whether there's either technology already available or it's of such high interest that we're going to help you. We'll figure out a way to do this. And these are the contact information, myself and Namrata, who is the associate director of the platform. And we start by arranging a meeting to discuss the science, and we take it from there. This is the group now, a little bit old photo, sorry about that, and some of our funding sources. I'll leave this with you, questions that proteomics can address, reviews on proteomic methods, tools, and technologies, and some additional resources. So I'm going to stop there and I'll field one or two questions, but I want to let the others get on with their talks.
So we're going to be tag teaming the next presentation. This is specifically about something called Immunopeptidomics. I'm Jenn, and I'm going to talk mostly about the wet lab piece and some theory behind what we do. And then after I kind of explain how we do these experiments, I'm going to be passing off to Mavi, who's our computational expert, and she's going to describe how we analyze this data type. So unlike a lot of the proteomics applications you may have been exposed to, immunopeptidomics is like a completely different world. So there's no tryptic peptides. There's no, traditional workflows, I would say, that everybody uses. This is kind of an area, even though it's been going on for, since the early 90s, it's an area that's still in rapid development. So specifically, most of our collaborators who want to do what we call immunopeptidomics or identify peptides that are bound to HLA molecules on the cell surface of different cells, they're interested in class 1 HLA molecules, and that's what's shown in this pathway. So unlike most proteomics applications where you're taking a whole cell, all of the proteins in the cell, you're digesting them with some enzyme with known specificity, what we're doing here is we're leveraging what the body already does with this pathway. So what happens is that proteins within your cell are constantly being turned over by the ubiquitin proteasome pathway. And peptides from this pathway get loaded onto class 1 MHC or HLA if you're in human molecules. These get shuttled to the cell surface for display, and TCRs in your body can basically go around and read these like billboards to tell if a cell is healthy or not. And we leverage the proteomics technology. So we leverage mass spectrometry and a bunch of biochemistry to actually isolate this population of peptides and sequence them. And this is really interesting because antigen identification is a really hot field, especially given the I would say, new interest in vaccines, which wasn't in there before COVID, as well as a lot of immunotherapies that rely on antigens being processed and presented. So there's other methodologies to identify HLA-bound peptides and characterize them. And one of the earliest was actually making one MHC molecule at a time, synthesizing hundreds of peptides and asking, does this peptide bind or not? Even though this is a good technology to kind of give you the general understanding of what peptides bind to each HLA, and I should also mention that this is the most polymorphic protein in the human genome. Each individual person actually has 6 unique versions of this protein, and each HLA or MHC has its own peptide binding motif, and there's over 20,000 of these. So it's a huge space to try to understand and cover. And so can you imagine trying to do an assay like this and characterize all MHC molecules, the peptides that are bound to them, and learn the rules of just binding? It's really hard. So in the early 90s, I would say this is when this field started. What happened is people were like, well, why don't we just take the cell and we isolate using antibodies against the MHC peptide complex in a very specific way, isolate the complex, remove the peptides from the complex, and sequence them by mass spec. That would give us a really rapid readout of which peptides not only can bind, but are actually processed and presented. Because there's a lot of rules before a peptide can actually bind of, does it have the right sequence context? Is it in a region of the protein that's easily accessible? Does it actually make it through this pathway? And you kind of can learn all the rules by just querying on a cell what is there. This is great. But because of what I just mentioned about each individual can have up to 6 HLAs, when you get this mixture, you don't know which peptide binds to which HLA or MHC that's present. And so to really understand that process, we've thought a lot about breaking this down even in a more simple form. Instead of looking at a cell that has all of its endogenous HLA molecules, we take a cell that doesn't have any, and we put in one at a time. And this really allows us to cleanly learn the rules of each HLA, and we can start to rapidly fill areas that there was no data. Because again, another thing is genetics. So a lot of the data in this field was really AO2 or the most common HLA specific. And if you want to serve a diverse population, you have to understand all of the binding characteristics of many HLAs. And so this technology allows us to just rapidly scan like the 100 most abundant HLAs very quickly, which is very cool. Just briefly, how do we do this in the lab? So we can take cells and we, depending on the cell type, almost all the cells in your body will have HLA-1. Really specialized cells have class 2 HLA. you can isolate them with antibodies against these complexes. Once you have the complex, the MHC and the peptide bound, you can bump the peptide off with acid and separate the complex from those peptides. And we introduce just those peptides that were bound to either HLA-1 or HLA-2 into a mass spectrometer and sequence them. And as I said previously, this single allele approach is a really clean way to start to learn the rules. And we have spent, I would say, about 10 years now trying to do this. And what I'm showing here are 2 publications in this area where we specifically take an HLA null cell line. We introduce 1 HLA at a time, and we're just learning the rules of not only binding, but also these processing and presentation rules. And the goal of this is to build machine learning predictors that we can use in the clinic to make personalized vaccines. Because it's really hard to have enough patient sample to do this on the fly quickly enough to generate these personalized approaches. And so all this data is giving us lots of biology, but it's also giving us this opportunity to make these machine learning models that are actually being used in the clinic today, which is really cool. And I will just say, again, we're learning the rules of which peptides can bind to which HLAs. We're also learning other rules about which proteins would be more likely to be processed and presented, and which contexts of a protein are really cleavable, which is cool. And we've done this for about 95 different alleles in this one system. And this data has been used quite extensively to build predictors, one of which is in-house built. It's called HLA Athena by CC Sarkisova. And what's great about this tool is it's publicly available. You can run it on Terra. It has lots of good functionality. So if you're interested in the HLA space, you can actually put in peptides or a protein of interest, and you can ask, does it bind to allele X? And you might be able to move forward with your assays, which is very cool. And the other thing I will note is we're not perfect. So if we could explain with this data everything about why a peptide could be presented, that means we would have 100% accuracy. We don't. We have like close to about 60 to 70% accuracy. So we still have room to go here in terms of learning these rules, which is also why sometimes we just need to do the real experiment, meaning we actually need to take cells, isolate the HLAs, see what peptides are there, and then we use that data to continue to refine these predictors. So I talked a lot about class 1, and class 1 is of high interest because there's a very clear, CD8 cells are killer cells. Class 2, at least in cancer and other diseases, isn't always as clear of, do we want to target this epitope? And so we asked the question, well, can we use the same single allele approach that we've been using for class 1 for class 2? And this actually ended up being much more difficult because there wasn't really a cell line that was class 2 null and had all of the other components of the processing pathway to allow us to be able to do that approach that I showed you previously. So we had to go back to the drawing board and design a new strategy, which ends up being really useful for lots of things, and I'll explain. So instead of taking a cell line that's HLA class 2 null, what we do is we take a cell line, any cell line we want, and we actually just put in a plasmid that codes for the HLA class 2 of interest with an affinity handle. And that will allow you now to generate the same single allele data without having to go through manipulating the pathway, which is really, really powerful. And this allowed us to basically do the same process. So in this context, what we're doing, instead of an HLA class 2 null line, we can pick any cell background we want that's transfectable. And we can go in very quickly within a 48-hour transfection, put in whatever the HLA class 2 we want with a tag, pull, get the data. This is scalable, 96-well. You can visualize how quickly you can fill in data here. And that's exactly what we did with this first publication. We looked both at class 1 and class 2, showing this affinity strategy is really useful for both. And we have since updated the use of this. So we started with this biotin acceptor peptide tag, which is great, but it's expensive because it requires BRA, and biotinylation is a little dirty for the isolation. So we have since redesigned this with help of Dan Graham and Ram Xavier's group, and specifically Martin and Jihei, which are co-authors on this. And we now really like the strep2 tag for the same approach.
A little bit about the physical characteristics of an HLA class 2 versus a class 1.
Yeah. So class 2 molecules, they bind different peptides, not only because the pathway itself is distinct, but because the peptides are really long. So for class 1, the peptides are 8 to 11 amino acids. For class 2, these are usually between 12 and 25, can actually go up to 50 amino acids. So they look totally different than tryptic peptides, actually both class 1 and class 2. And class 2 molecules are much more complicated than class 1, because in a class 1 space, what happens is you have an alpha chain and B2M, which is not variable. With class 2, it's a heterodimer. You have an alpha chain and a beta chain that come together. For DR, one of the alleles, the alpha chain is really monomorphic. But for DP and DQ, they can pair in multiple ways. So instead of having 6 HLA1s for class 2, you can actually have up to 12 combinations. And there's also some like genomic linkages with DR. You have DRB1 linkages with DRB345, and that's what gets you the number of 12. So it's just like crazy complicated for all of those reasons. The other interesting thing about class 2, which is distinct from class 1, which actually will help us in a data analysis perspective, is that for class 1, you're usually only getting 1 or 2 peptides per source protein. But in class 2, because of the structure, what happens is you get the peptides in what we call nested sets. So you get like 3 or 4 peptides from the same protein that all differ by 1 amino acid. So you get the 13 mer, 14 mer, 15 mer. So actually the data types between these two are really distinct. So not only are the isolation methods different, but the way that we acquire the data has to be different, because the peptides look different in size and length and all these characteristics. And as Steve mentioned, we've worked really hard on this protocol in isolation, but we have since figured out how to basically put the immunopeptidomics protocol with all the others, and Shanka will talk about this and how we're using this, what we call Monty protocol, to be able to take one sample, be able to isolate the HLA peptides, and then pass the flow through of that to do all of the other omics. So this, I think, is going to be very powerful for a lot of different studies. And what that will allow you to do is not only understand what's actually processed and presented, but you can actually go into the data and start to look at some of the genes that are involved in the pathway. And what I'm showing here is B2M, which is a component of HLA class 1, and CD74, which is a class 2 component. And this might help us see, like, if you have a cell line, the HLA repertoire seems to be lower, we can actually go and look at the pieces of each of those pathways to try to understand why a certain cell line might not be functioning well in the antigen processing presentation scheme. So we generate lots of data. I've hinted that the data is very complicated. So we've had to put in a lot of time to build tools to enable the research in this area. And I'm going to pass it off to Mavi, who's going to describe some of these tools.
Good afternoon, everyone. So I will be presenting the computational methods that we are using here in the proteomics group for the analysis of the immunopeptidome. So here in our group, we are interested to identify tumor-specific antigens for the development of T-cell focused immunotherapies. So the immunopeptidome is the repertoire of HLA peptides that are presented at the cell surface to mediate the antigen presentation. So to those peptides that the T cell react and trigger immune response, we call it antigens. And in cancer, we have several types of antigens. One kind are the neoantigens, that are peptides that derive from the mutations in DNA sequence. Therefore, these peptides are solely presented in tumor cells. They are not presented in the normal cells. Unfortunately, identifying these peptides is a bit difficult. That's why there is only a few number of these peptides that have been identified so far in most cancers. But when we can identify them, we can develop personalized immunotherapies because these peptides tend to be patient-specific. Other kind of peptides are the aberrantly expressed antigens. These peptides derive from the aberrant translations that are specific to tumor cells. One example of these are the cancer testis antigens, or CTAs. These peptides derive from the aberrant translation of some genes that are presented in the tumor cells, although this can also be presented in normal cells, but because these cells don't have the presentation of HLA, we can take these antigens and develop personalized or immunotherapies to target the tumor cells. Other kind of peptides are the non-canonical antigens. These are peptides that derive from aberrant translations that are also specific to tumor cells, that derive from supposedly non-coding regions in the genome, for instance, the intronic, intergenic regions, and also from the untranslated regions of canonical genes. In contrast to the neoantigens, the variantly expressed antigens tend to be more abundant, and they also have been seen that they can be highly shared between patients. So this means that from a single peptide, we can develop an immunotherapy that can target several tumors. And to be able to identify this aberrantly expressed antigen sequence, we can develop or use, for instance, ribosome profiling data. So the ribosome profiling is a technique that allows or helps us to identify those actively translated regions in the genome. The ribosome profiling has three main steps. The first one, we need to use a translation inhibitor to block the ribosomes onto the mRNA molecules that they are translated in vivo. A second step is the footprint recovery. This comes after the lysis of the cell and the use of enzyme, that is the RNase, that is going to cut the mRNA molecules that are not protected by the ribosomes. So at a certain point, the ribosomes then are going to be removed from these mRNA molecules, and we end up with these very short fragments that were protected by the ribosomes. With this fragment, we do the library preparation, and then from this library preparation, we can do the deep sequence to get the mRNA sequence for these protected fragments. Then we can, as a result, we have the ribosec profiling reads that we come up onto the genome, and from there we can use prediction algorithms for detecting the novel or unannotated open reading frames. So in this example, using this ribosome profiling data, we discovered a short protein in the 5' UTR of this canonical gene, the MLH gene, that has the translation support from the ribosome profiling data. So as we are in our search for antigens, we need to include these novel or unannotated open reading frames into the database that we are going to use to make the identification of the HLA peptide sequence. So for this reason, we include the nuORF database, that is a database that contains non-canonical open reading frames that were already detected using ribosome profiling in 29 samples, including normal and cancer samples. So this database contains a lot of thousands of non-canonical open reading frames that have been observed from these supposedly non-coding regions in the genome that include non-coding RNAs, pseudogenes, 5', 3' UTR of canonical genes, and also out of the Fresno translation of protein coding. So to be able to identify all this variety of antigens, we need to include the reference proteome, which is the gen code 42. So we are able to identify the cancer-testis antigens. We then add the nuORF database to identify the non-canonical antigens. And finally, we also include patient-specific databases to be able to identify neoantigens. So for this discovery of the patient-specific database, we use, for instance, whole exome sequence or RNA-seq data to find the somatic mutations of aberrant spliceoforms for the samples that we are analyzing. So this is the general approach that we use to identify and to validate the tumor antigens. So in our first step, we need to identify the HLA peptide sequence. And in a second step, we are going to narrow down this sequence to find those that are most likely to be tumor specific. So for this first step, we collect our cancer samples. We do the immunoprecipitation. We collect the peptides, so those that are bound to the MHC1 molecules, and we inject those peptides in the mass spectrometer. Then we do the identification of the HLA peptide sequence using the database that we constructed with the canonical and non-canonical proteome. So at the end, we have for each one of the samples the list of peptides that were presented on the cell surface for those cells. The second step, in the validation step, we need to filter in those peptides. So we have several filters. And the first one is we are going only to retain those peptides that have a strong binding affinity between the peptide and the HLA molecule. So at the end, we have a narrow list of peptides that then we want to look at their expression in normal and cancer samples. And we are going to retain only those that are most likely to be cancer specific. At the end, we have a very short list. And from then we can, for instance, do immunogenicity assays to evaluate how able these peptides are to trigger the immune response from the T cells. And at the end, we end up with a list of selectable targets that can be then used for the development of an immunotherapy. So for this first step of validation, to look at those peptides that have good affinity between the peptide and the HLA allele, we use the HLA Athena, which is the model that Jenn just explained before. And for the prioritization, we need to be very careful because we want to evaluate the expression of these peptides in RNA-seq data. So we need to be very exhaustive and evaluate the expression of these peptides to be able to do a fair comparison between the cancer samples and the normal samples. And to do this comparison, we are going to look at the expression in normal samples. So we are going to include all the tissues, the normal tissues from the GTX data. We are going to retain those peptides that lowly express in normal samples. And from there, we are going to evaluate their expression in cancer samples to retain those that are highly expressed. And to look at this expression of these peptides, we need to be very exhaustive. So we are going to evaluate the expression in all the genomic locations of the peptides. Because it could be possible, for instance, if we decide or we choose or select only one region as the source of a peptide in cancer cell, if we compare that region to that of normal, we might have a biased comparison. So we are going to collect the expression in all the genomic regions to be able to compare it with normal, because it could be possible, for instance, that in normal, there are other regions, including those that have been annotated as non-protein coding genes that can also code for the same peptide and result in a high presentation of the peptide at the cell surface. And to facilitate assessing this HLA peptide expression, we want to use BanQuery, which is a tool that allows for each peptide get the expression in any RNA-seq sample. So BanQuery needs two inputs. The first is the list of peptides, and the second is the band directories. And the band directories is the list of band files. So the band files are those files that have the line reads for the RNA-seq samples that we want to evaluate. And then from there, BanQuery use or perform 5 main steps. The first one does the reverse translation. From the amino acid sequence, BanQuery collects all the nucleotide sequence, and then use this nucleotide sequence to map all these sequences on the genome. The next step is to query each one of these regions of the genome in each one of the band files to get the number of RNA-seq reads that are mapping to each location. And the normalization step is going to sum all the RNA-seq reads for every peptide in all the locations. It's going to sum up those reads, and it's going to normalize those counts by the total read count in each band file. So at the end, we have the expression of each peptide, whether in the cancer or normal samples, and we can do the comparison of the expression in the RNA-seq data. The final step is the biotype classification that will tell us in BanQuery from which region of the genome I found the reads for every peptide. So using BanQuery, we wanted to evaluate those tumor antigens that were already previously identified in previous studies, and we wanted to compare or look how good it is to look at the prioritization of those peptides. And in here I am showing an example of a neoantigen that was previously collected or identified by Immunopeptidomics in the colorectal cancer. So we compare the expression of this neoantigen to the expression of the wild type in all the normal tissues from GTX. And as we can see here, the expression of the wild type is very high in all the normal tissues, while the expression of the neoantigen is very low or is non-existent in these tissues. So this suggests that this peptide might be a good target for the development of immunotherapy targeting the colorectal cancer. Moreover, we collected other tumor antigens that were already used in clinical trials, and we wanted to look again their expression in the GTX data. So we wanted to look if with BanQuery we might be able to predict the outcome by using these peptides in an immunotherapy setting. And in the top of the heat map, we have 5 of the antigens that we look at the expression, and we see that they have very low or no expression at all in the normal data of GTX. And from these peptides, they didn't report any adverse events. However, for the others, we can see that they have more expression in the normal tissues. Some of them, they are very high expressed in those tissues. And for these peptides, there were more safety concerns and also some extreme adverse events. So for these two here, they present some safety concern, although they are not extreme. Some patients that were applied these immunotherapies, they present anorexia, hypertension, vomiting, and rash. For other types of antigens, some of these patients present more extreme events as cutaneous events in skin skin and eye autoimmunity. Others present severe colitis, cardiac events, and also severe neurotoxicity. So what this says is the PanQuery is a great tool to help us to validate the antigens and to find those that are more likely to be cancer specific. So we are going to prevent that using these peptides in an immunotherapy setting is going to trigger some adverse events. And with this, I want to do the summary of this presentation. So we see that the immunopeptidomics is a powerful tool to understand the antigen presentation in the context of disease, in particular in cancer. Also, we see that the data we can obtain using limited amounts of patient sample continues to increase. This is because we are constantly working in the optimization of workflows. In the other, sensitive and accurate mass spectrometry immunopeptidomics approach are required to robustly identify non-canonical and neoantigen peptides. So we are not relying only on prediction tools that might lead to false discoveries. And we see that using the optimization of the mass spectrometry search strategies, using ribosome profiling data, for instance, can help us to identify more confidentially the HLA peptides. And also using PanQuery, we can see that we can prioritize those antigens that can be used as good targets immunotherapy for the immunotherapy. So PanQuery can be like a first validation step to prioritize those peptides before to do other types of validations. As some future directions, so we are currently analyzing hundreds of samples on melanoma, sarcoma, and chronic lymphocytic leukemia. So we're hoping to discover a lot of antigens that can be helpful for the development of therapeutic options for these kind of cancers. And finally, we intend to exploit all the data that we're generating in our studies to use this learning for learning more about peptide translation and presentation that hopefully will help us in the future analyzing other types of cancers. And by this, let me thank for all the members of our team, our collaborators, and the funding agency. Thank you.
So when you're predicting the activity of the HLA peptides using HLA, first you take into account if the peptide has a PTM. Can it predict still with a PTM?
Yeah. Yeah, and the question is, does the prediction tools, and this is true for all predictions, right? And it predict if the peptide has a PTM in it?
Yes.
And the reason is so as Steve says, like PTMs in general, are there most of you like an enrichment strategy. And for us, it's really hard in the HLA world to use those same enrichment strategies because the input level of peptide we start with is like 100 to 1,000 times less abundant than what we get like a tryptophan test. Doesn't mean it's not possible if we've done it at a really low throughput. And so what that area really needs is really large training data sets that we don't get. So we have the biggest training data set for phospho, which is one of the more abundant PTMs, is only a few thousand identifications.
So another question I had was, can you also acquire data for in the mass spec using DDA and DIA both approaches and both work? Or?
So Claudia is actually working on the DIA approach now. The caveat with us is that something I maybe didn't make clear is for tryptic proteome searches, you can tell the database to digest all those proteins with a tryptic specificity because there's an enzyme. For HLA, you don't know what the specificity is. And so you have to basically take the whole database and tile it for every possible peptide, which creates a database space that's 100 times larger. And for us to be able to confidently identify a peptide using the DIA approach, we are building a new set of tools that include a strategy that's being worked on in Near's lab, which will allow us to actually generate the synthetic standards in a high throughput way. So we actually have a measurement of what the real peptide is that we can use to deconfirm our DIA data. So that is for sure something we're excited about, but we're not there yet because of that database search issue.
Thank you. I have a question as well. How do you deal with cysteine-containing peptides that get displayed extracellularly? So I did try to alkylate them with the stomach, or they're oxidized already.
Yeah, so I think the question, and I just want to make sure, is cysteine peptides.
Yeah, cysteine.
Yeah, this is a problem. So historically, reduction in alkylation wasn't used in immunopeptidomics data processing. And that's because, again, we have really low levels of material, and that step causes loss. And so a lot of the historical data doesn't have reduction or alkylation because you lost so much of your peptide trying to do it that it wasn't working. So you would just get whatever the version of the cysteine was. And one of the modifications that's common is something called cysteinylation, which you actually have a free cysteine binding there, is something that we can see in our data. Oxidized cysteine is much more rare, we rarely see it. And unfortunately for us, a free cysteine doesn't fragment. We have since improved the protocols. So now we can either include a alkylating agent in the lysis buffer. So basically any free cysteine, it'll add that too, so we can see it better, but it will maintain the cysteinols and some of the real like PTMs that might be there, modifications. And we can also completely reduce now.
All right, if no more questions. So my name is Shanka Satpathy, and I'm a senior group leader in the proteomics group. And I will talk a little bit about the work, our work in the area of application of proteomics. So I engage both in technology development, but I'm also very interested in applying some of the tools we develop in-house to biological questions. So you have seen this slide, but essentially a big focus of what we do in the cancer domain is cancer proteogenomics, or multi-omic analysis covering many different types of cancers as listed here, and many more that we're working on. But in brief, essentially from the same tumor, we are obtaining genomic information, different types of genomic information, starting from mutations, mRNA expression, microRNA copy number, and epigenetic modifications in some cases, for example, DNA methylation. And then we are also from the same tumors trying to do a bunch of different proteomic analysis, for example, looking at whole global protein expression, PTMs such as phosphorylation, glycosylation, acetyl, and ubiquitylation. And also, as you heard, we have also extended this to including to include immunopeptidome. And at the end of the day, once we have all these different disparate data sets, the logic is essentially to integrate some of these data sets and to investigate tumor biology, or to understand tumor microenvironment, identify new drug targets, for example. And as I said, we have applied this to many different types of tumors, some of which, and we have extensively published reviews around that as well, that you can read. This slide also you have seen before. Essentially, the idea is we can start with 25 to 50 mg wet weight. A lot of the projects that I talked about or I showed previously, cancer tissue types, the work was done with surgically resected material. So we start with, as I said, 25 to 50 mg wet weight, and then we lyse it in a native lysis buffer. And then HLA-2 and HLA-1 immunoprecipitation is performed, and then the flow through is denatured using SDS, digested, and then we we can do PTM and proteome analysis in a serial fashion. So first, we start with ubiquitylome, and we use TMT, tandem mass tags, and DDA-based approaches to pool up to different 18 different biological samples, so that and analyze that in a single experiment. And we take the flow through from these ubiquitylome analysis, and then we fractionate it to reduce the complexity, and we do take 5% of that for full proteome. We enrich for phosphoproteome, as you heard before, and we also enrich for acetylome. And all of this is done, as I said, using tandem mass tags. These are essentially barcodes that attach to your peptides, and you can pool up to 18 different samples, and all of that can be done in a single experiment. So in essence, all of this is happening from a single tumor source. And depending on the biological question, you can also do in a modular way. You can do the proteome only, you can do the phosphoproteome only, you can do the acetylome only, and so on and so forth. So now I'll dive into a couple examples of what we have published and how proteomics has added value to some of the extensively studied cancer types, primarily by genomics until now. So this is a study we published back in 2020 2020, where we did proteogenomic analysis of lung adenocarcinoma, or LUAD. And here I'm showing proteomics data scatter plot compared to RNA. So on the x-axis, you have fold change between EGFR mutant tumors and EGFR wild-type tumors. And on x-axis, you have RNA, and y-axis, you have proteomics, not only just global protein expression, but also phospho and acetyl color-coded differently. And as you can see, one of the key outlier site that you see is PTPN11, which is also known as SHP2, a very important protein downstream of several receptor tyrosine kinases. And the Y62 is supposed to activate the protein. So from this plot, it's certainly clear that in EGFR mutant samples or patients, PTPN11 is highly phosphorylated. That marks as a site for activation. And some of this information is completely remissed if you just were to do genomics. And for the same protein, we see a slightly different trend when we are looking at ALK fusion samples. So this is also a recurrent fusion that you see in LUAD. And you can see the same protein is phosphorylated, but on the C terminal, so completely different. And the biological consequence might be different as well. So these are kind of information that you can glean from PTM data. And this is another example where we take all these different layers and then we try to integrate this to get into the immune oncology question. What's the immune landscape within these tumors? And then there are, I don't want to go through the entire heat map, but essentially there are proteins and pathways that is exclusively captured in the proteomics data. So these are all the pathways that have been exclusively captured in the proteomics data, for example, and have not been seen in the RNA. And one example is what we found in the STK11 mutant samples. So STK11 mutation is also recurrently mutated, recurrent mutation in lung adenocarcinoma and is associated with cold phenotype. And you can see here, this is a fold change expression analysis between STK11 mutant tumors and wild type at the level of RNA and protein. So all these different bunch of proteins that are upregulated only exclusively at a level of protein expression, and all of these proteins correspond to a single pathway called neutrophil degranulation. So based on this, we saw that neutrophil degranulation pathway is enriched in STK11 mutant tumors and is exclusively shown or highlighted from the proteomic data set. So this is another example that we published a couple years back in LSCC, or lung squamous cell carcinoma, another form of lung cancer. And so as you can see here, FGFR1, which is a targetable receptor tyrosine kinase, is recurrently mutated in a large subset of LSCC, or lung squamous cell carcinoma. Our data showed that it's not FGFR from this amplicon, but NST3, which is next to FGFR, is overexpressed from this amplicon. And there have been studies where FGFR was the focus, but apparently none of those clinical trials worked, and a primary reason was because FGFR was not amplified and was not overexpressed and was probably not a vulnerability. And while our paper was under review, there was another paper that showed the role of NST3 in LSCC carcinogenesis. It nicely validates our study. So, the kind of studies we often do and should be applicable to lots of different tumor types. We profile tumors. We also profile adjacent normal tissue when possible, and then we can essentially identify proteins that are regulated in tumors exclusively, and we can bin them into different categories: druggable kinases, ubiquitin ligases, E2s, and so on and so forth, enzymes. And then we can also associate some of this expression with critical features, for example, progression-free survival, overall survival, relapse-free survival, and all of that information when available. And then we can also take this data one step further and ask the question if some of these proteins are inherent dependency. And we can also mine different databases, for example, DepMap, as we have done in this paper. So I did mention a lot of the studies are surgically resected tumor, but a lot of the clinically available material might not be surgically resected tumors. So doing some of these studies is tricky. For example, we typically work with biopsies and also FFPE, as I'll talk about. But we have developed a method that we call biopsy trifecta extraction, where we can do proteogenomic analysis from a 14 to 18 gauge needle core biopsy. So what we do is we take a biopsy and then we take interspersed 5 micron IHCs to make sure it's tumor rich and it has sufficient tumor in these biopsies as you move through the biopsy. And then we take alternative 50-micron section, we pull into 3 different tubes. The first tube, we can enrich DNA and protein. The second tube, we can extract RNA, and the third tube, where we can extract non-denatured protein for things like IPs or kinase inhibitor pull downs. And the depth that we get is reasonably, reasonably good. So we can identify, more than 11,000 proteins and 70,000 phosphosite. And some of the work we have done in this area also leverages TMT DDA-based approaches. But the fun is actually in applying it, and we have applied it to a bunch of clinical trials. This is one of the latest papers that we have published in triple-negative breast cancer, where patients where triple-negative stage 2 and 3 patients were given chemotherapy, primarily docetaxel and carboplatin, and biopsies were collected pre and post-treatment. And we have done, and these are all OCT embedded samples, and OCT can cause significant problem in mass spec. So we have devised a way to clean the OCT very thoroughly, and then we can really get good mass spec data. And so in this study, in this study, using both genomics and proteomics, we are able to identify meaningful biomarkers for chemo response. For example, in this study, we identified Lig1 or a DNA ligase 1 loss as associated with disease progression and chemo resistance. And we have also validated some of this information in PDX organoids, where we can show that loss of Lig1 is associated with carboplatin resistance in this case. And we also engage with our collaborators in doing extensive follow-ups, and there is so much fun in these types of projects. For example, in this study, in this aspect of this figure, a patient who was given a response and also was given the chemotherapy and was also part of the study, initially responded. So PDX model was made out of her biopsy. Then the patient developed resistance and subsequently metastasis. So 3 biopsies were taken, and then PDXs were generated, and then some of the treatment experiments were done in PDXs. As you can see, as the patient is responder, non-responder, metastasis, you can see a progressive loss of this Lig1 protein, which was very interesting to see, and also good validation of what we found. So another form of cancer tissue that we have access to, and a lot of people have access to, and is probably current in pathology lab, is FFPE. These are not the best samples out there, but these are the samples that's out there, especially for which you have extensive metadata available tens of years. So we have developed workflows to be able to get very good proteomic depth from these FFP tumors. So essentially, in this protocol, we take a single block, we take a 10-micron scroll, and then you can macro dissect or micro dissect the tissue to enrich for tumor, and then we transfer into a plate, and we have a semi-automated workflow that's in place where we do all of this processing, such as deparaffinization, decross-linking, homogenization, extraction, all of that in a 96-well format. And then there are automation steps and semi-automation steps from the point of where you lyse the samples to all the way to peptide cleanup and adding it to running it on the mass spec. And we have a bunch of different acquisition options available, and that will also dictate the depth of this proteome experiments. And one thing that I would also mention, we also are working, and we have access to cloud-based infrastructures where large-scale data sets can be analyzed in a reasonably seamless way. So this is, a lot of these proteomic analysis are done using DIA, and I don't want to repeat this, but Steve explained this quite elegantly, that DIA has its strength. You don't need extensive fractionation, and the data completeness is far better than DDA. And also, because there is no fractionation and you can do it in a label-free way, you can also automate this, but also, apply this to large-scale projects and get data very quickly. So these are some, I don't want to belabor on the DIA methods, but the point of this figure is the sample prep requirements for DIA is significantly shorter than extensive TMT-based workflows that requires fractionation. So the sample prep is significantly shorter. You can do it in 96-well format. The data, the number of data points, as in protein in this case, you can get from 7,500 to close to 9,000 proteins, depending on which platforms we use. We have, I have listed 3 different instruments: Exploris, timSTUF, and Astral. I don't want to go into detail, but depending on the instrument, you can also reduce or increase the sample per day throughput. For example, this is 48 sample per day, and per sample you're getting 9,000. We have some methods, 12 sample per day, you're getting 7,000. So as the field is evolving, the instruments are also evolving, and the throughput is also evolving, the depth is also evolving. And this coefficient of variation is also quite good for these sorts of experiment. And here, essentially, I'm plotting the data quality. So in this case, we have taken 2 FFP blocks, one that overexpressed a protein of interest, and another one was wild type, and essentially I'm just plotting the fold change. So, and on the x-axis, this was a DIA data acquired on Astral platform. I don't want to go detail, but it's an instrument essentially. And then comparing it to TMT DDA, DIA on timSTUF, another Bruker system that's available, and Exploris, which is another instrument. But the point here is, no matter how you slice and dice it, there is a very good data quality and very good reproducibility across different platforms. And the good thing is it's fast. But this DIA-based workflows are not exclusively to tumors, not exclusively to FFP material. We can apply to cell lines, organoids, PDXs, and any other biologically relevant material. And this is one of the examples that Steve talked about, where we are engaging with the DepMap team to profile a large number of cell lines. And as you can see here, we are getting close to 8,000 plus proteins from a single cell line. And here I'm plotting for a subset of these cell lines we did in triplicates, and as you can see, one protein is plotted here. The coefficient of variation is wonderful. I talked a lot about cancer biology, but we are not exclusively focusing on cancer biology. We have a lot of different kinds of projects in the group. We have projects on neurobiology, infectious biology, and metabolism. And this is one of the interesting projects that came out of your multi-omic project that came out of our lab recently, where these rats were subjected to chronic exercise training, and then they were sacrificed and different tissue were extracted, and multi-omic analysis were performed, and this is part of this MotorPack consortia. And as you can see here, and in this heat map, essentially, a lot of the differentially regulated features are plotted, and as you can see here, different omics is giving you like different information, and so which is the strength of doing these multi-omics analysis. And finally, with so much data, what do we do with so much data? So Mani is probably going to talk about as well. Once we have all this data, we are also investing in thinking very hard on how to analyze the data, or how to provide easy format to not only visualize that data, but also understand the data. And then there are cloud-based platforms that are being built to take all these different omics data and then come up with biologically relevant features or interpretations. And to summarize, I talked a lot about our engagement in the multi-omics and proteogenomics front, and both in tumor biology, but beyond that, where we are engaging different collaborators, different groups, different cancer types. I also showed a little bit data on how we are using proteomics to investigate response and resistance in therapies, some of which might not be apparent exclusively in genomics. For example, the HLA immunopeptidomic analysis has added this wonderful dimension to tumor biology and what we can really learn about immuno-oncology, for example. As we think about different projects, we are inventing and optimizing existing and new proteomic technologies to make clinical proteomics really as robust, as reproducible, and as fast as possible. And finally, with all the data, we need sophisticated computational biology tools, and our group is really invested in that as well. So with that, I would like to thank, again, this is an old picture. Some of the members are missing here, but overall, I would like to thank the wonderful BroadE Proteomics team and funding sources. Thank you.
Okay, so now let's take a step back from application more to technical development. I will be mostly focusing today on our efforts, how we push sensitivity limitations of current mass spectrometry-based approaches to achieve deep proteome coverage of 100 or even down to a single cell. And the first question is, why do we even need single cell resolution? And what I think we have seen from very successful transcriptomics approaches is that tumor tissues, for example, are basically any tissue is made out of a lot of different cells with different functionalities, and they're present in different ratios. And if we would analyze that tissue on the left-hand side in bulk, we would mask this heterogeneity and therefore also not be able to distinguish these different cellular identities and what actually gives rise to healthy or diseased tissue. Additionally to that, we're also very interested in the spatial context. So not just the single cells per se, but actually how cells influence each other by being in spatial proximity or even by expressing more signaling molecules towards each other. Now, in order to enable single-cell or spatial proteomics, we are faced with 4 main challenges. And number 1 would definitely be sensitivity. Most proteomics approaches that we have heard and will hear today are based for about 10 million or even more cells of input, especially when we are looking at these Monty approaches that we have heard about. We're dealing with strong adsorptive losses. So for example, a single mammalian cell carries about 150 to 300 picograms of protein. And we really need all of these, all of that to reach our mass spec in order to achieve a sufficient signal. We're also challenged with reproducibility, where we need to distinguish or minimize technical variance and really preserve the biological variability that we're so interested in. And lastly, the throughput. Most proteomics approaches are either relying on a lot of in-depth fractionation, or even if we're doing our single-shot experiments, we're usually dealing with around 1 hour of acquisition time. However, when we look at single-cell analysis, in order to achieve statistical significance to really describe what that tissue is made out of, we're looking at 100, 1,000, or even more cells. Now, we have developed specific workflows that start with either tissue blocks, we're here talking about cryo or FFPE blocks, and cells in suspension. For the first, we're using, as Steve was briefly introducing, laser capture microdissection to really select out subsections of those tissue blocks. And for our workflows for cells in suspension, we sort out our cells of interest or our cell population of interest, and then subject them to our very dedicated workflow. That includes cell lysis, protein digestion, and then highly sensitive LC-MS/MS analysis. And I will start off with our workflow for our single cells in suspension, where we're really relying on one specific instrument that we have in the lab, and that is the Cellin-1. And the Cellin-1 is an acoustic-based precision dispensing robot. We use this glass capillary that you can see on the right-hand side to generate picoliter-sized droplets at extremely high speed. And when we look closer how these nozzles are based, we can also see that we can use this for image-based cell isolation. And we can see here, one of our cells is moving through this glass nozzle. We distinguish between the ejection zone and the safety zone. And in order to make a little bit more sense out of this, when the instrument detects a particle that matches user-defined parameters such as cell size, granularity, and fluorescence, that particle is being isolated. That particle is being isolated and actually deposited in your location of choice. However, when the instrument doesn't detect a particle in these 2 zones, or a contaminating particle is present, these cells are not being isolated. They're actually deposited in a recovery well. So if you're working with a very, very rare cell population, you can take them up again and go for a second round. Now, the entire robot is also temperature and humidity controlled, so you don't really have to move back and forth between different instruments, but all of your incubation steps and the entire pipetting is performed within that robot. And lastly, that also allows for workflow automation. So since we're working very, very close with this company, they have come up with automated workflow where you basically press a button and the entire software will guide you through incubation steps, quality control steps, and basically allows you to perform that very standard workflow. If you're looking to do something more fancy, that is also possible, not as automated. So in close collaboration with Selenium, we have come up and we have developed a chip-based workflow. And that chip is, we call the ProteoMics chip EVO 96. And it's basically in the layout of a 96 well plate, and it is made out of very low peptide absorptive Teflon. And it consists of these very specifically shaped wells that you can see right here. And what we do in our workflow, we're actually using a very specific oil. That oil solidifies at below 18 degrees Celsius, and we use it to basically generate a pedestal in these wells. Now, using our Cell-in-One, we start to dispense very little volumes of what we call the master mix. That is a combination of the detergent for cell lysis and the enzyme of your choice to perform proteolytic digestion. Then we deposit the cells, as I was introducing earlier. We add another round of master mix just to make sure that our cell is really nicely engulfed by our master mix. And then for incubation, we increase the volume. We increase the temperature. That increase of temperature also melts the oil and basically engulfs the entire droplet of master mix with the cell and overcomes evaporation throughout prolonged incubation times. And this very dedicated workflow is actually approaching all 4 of our main hallmarks of sensitive mass spectrometry analysis. Number 1, we're using the latest generation mass spectrometry instrumentation that we have here available at the BroadE. And our dedicated workflow combining the ProteoMics chip EVO 96 with the Cell-in-One actually allows us to overcome a lot of these technical variability that we're experiencing with, and the miniaturization of the workflow really allows us to recover as much protein as possible from these single cells. And with this very streamlined workflow, we currently can process 96 samples in only 3 hours, which is very, very important because we're working with live cells. And we acquire 40 samples per day, which allows us to achieve a proteome depth of over 4,000 proteins per single mammalian cell. And that actually spans 4 orders of magnitude. That is not a very high number for the transcriptomics field, but just to put that in relation, we were starting with like 100 proteins 4 years ago. So this is really, really exciting for the single-cell proteomics field. And we really also wanted to see if we can recapture biology using this workflow. And for this, we are using THP1 monocytic cell lines. We treated those with lipopolysaccharide, or short LPS. And just to give a brief overview of these, of the entire workflow. In the previous slide, I was showing you the analysis of HEK cells. HEK cells are almost more than double as large as those THP monocytes. And this is especially critical when we're talking protein. Proteins cannot be linearly amplified. So if you have a smaller cell, you also have less protein. And that is actually reflected in the protein identifications that we can get out of those cells. From these very large HEK cells, we can easily identify around 4,000 proteins. However, when we're dealing with very small cells, as those at THP1s are, we're looking at an average around 1,000 proteins. So that was a little bit discouraging. However, we were looking into those expression profiles. And what we can see is that we can actually distinguish the THP1's monocytes that were treated with DMSO versus the ones that were stimulated with LPS, only looking at PC1 and PC2. What was the most interesting factor for me was actually that across these hundreds and hundreds of cells that we were analyzing in this case, we identified over 200 significantly regulated proteins with an adjusted p-value of 0.05. And we really wanted to know if those significantly regulated proteins do actually recapitulate what we expect our LPS treatment to do in those cells. So we were expecting that our monocytes are being activated to macrophages via stimulation of the TLR4, release of cytokines, activation of immune response via the NF-kappa B pathway, and activation of type 1 interference. Now, we took our 200 significantly regulated proteins and performed gene set enrichment analysis, and we're quite happy to see that we are actually seeing what we were expecting to see. And that is enrichment of proteins of interleukins, interferons, inflammatory responses, and also post-translational protein modifications, all of which were already heavily described in literature to be associated with LPS treatment in those cells. Now, moving away a little bit from our workflow from the single cells in suspension, we, as I said, also developed workflows for tissue blocks. Here again, we're working with both paraffin-embedded tissue and cryopreserved tissue. We excise these tissues and then perform basically the exact same workflow as I was introducing to you earlier. So if we go a little bit in more detail, our last workflow that we performed was we were receiving FFPE preserved patient tissue from a collaborator. We section 10 microns, 10 to 15 microns sections, and then we select different cells or cell areas. To make that clear, we're here not looking at single cells. We're trying to identify, and we're really actively pursuing in this area to identify cells that are similar in morphology or that expressing similar markers to be able to isolate 100 or 1,000 cells or cell areas that are distinct from the entire tissue that we're looking at and might be of interest. Now, we're using the laser capture microdissection to collect those hundreds to 1,000 cells in one of those wells that I was talking to you earlier, and then we perform our one-pot sample preparation. Now, with this workflow, we're again trying to address our four main aspects. Here, we're focusing at the sensitivity level, not just on instrumentation of mass spectrometry, but also newest generation of laser capture microdissection and a lot of different other instruments that we are very happy to have here at BroadE. And additionally, here we're again facing an additional challenge when we're looking at losses per sample, and that is sample preservation. Really, the FFPE sample preservation process cross-links proteins, as Shanka was introducing to you, and that reduces the proteins or peptides that we're able to recover from these very low abundant samples. Now, we're also trying to implement new software and also publicly available software to be able to distinguish better between biological and tissue quality differences. So to enrich for cells that are highly similar in that case, and select those out to boost our signal above the noise. That will also help us with our throughput, where we're currently actually manually selecting these sub areas of tissue, where we really want to have a more streamlined and more automated workflow to analyze, again, 40 samples per day. In the current workflow, when we analyze about 1,000 to 500 cells, we're achieving up to 3,000 proteins. And that is still an active area of development. So just to briefly summarize what I've showed you today is that our prototype workflow is aiming to scale current mass spectrometry-based approaches towards single cells or to preserve spatial context within tissues. We have automated sample processing in a high throughput manner that really allows us to achieve the throughput that we are aiming for to achieve these statistical significance. And with the dedicated instrumentation that we have in-house, we can identify up to 4,000 proteins from a single mammalian cell. Now, when we extend this to our spatial proteomics workflow, as I said, about 3,000 proteins from around 1,000 FFP preserved cells. And we're really focusing on integration, the automated selection of cells that are highly similar in morphology or expression of specific tissue markers. And we would really be interested in talking to you about any kind of interesting projects that you would be up to and interested in pursuing that technology. And with this, I'm already at my most important slide. Of course, the proteomics team, but also our flow core and everyone for their support. Questions?
What are your thoughts about combining the previous strategy with the single cell proteomics strategy to enable multi-omics, single cell analysis?
Yeah, so I think multi-omics analysis of a single cell would be the ultimate goal. That would be really cool. There has been a couple of, or let's say 2 successful approaches to do this by really splitting up the cell to be able to allow for proteome analysis and then looking at different ways how to recover multiple molecules or try to overcome the noise over trying to increase the signal over the noise in this case. But I think this is still very challenging. We are very interested in moving across to the PTMs for these very low input levels. This is also an area of active development, such as immunopeptidomics for a lot lower input levels, definitely not single cells. So yeah, that is an area of development.
To add to that, there are now active projects, not in our lab, but in other groups, doing single cell metabolomics. So that is actually somewhat easier than doing single cell proteomics.
What about single cell glycomics?
Single cell what?
Glycomics.
Well, first thing you have to ask is who cares? Past that, back into what exactly is the question to ask? Is it site of modification? That's certainly going to be doable because modifications sites are understood, hidden like sites, by removal. Okay, it's PNGAs. Okay, you end up with a site that is either has been converted from an asparagine with a carbohydrate on it to aspartic acid. And you didn't see it before. After PNGAs, now you suddenly see the peptide. But if you want to go after single glycoforms from a single cell, that's a tough question. Presumably you'll get some information. Question how deep, I don't know. It's not something we're actively engaged in.
Okay. Yes, well, hi everyone. My name is Malvina. I'm a scientist in STIPS group, and we are going to switch gears now completely, going from the proteome level to the single protein or complex level, and talk about hydrogen deuterium exchange mass spectrometry. As the title indicates, HDXMS is a technique for probing protein dynamics and interactions. And the technique has been out there for many years, and we have seen many technological advances over the years. But some of the typical things that you can do, and I'm showing here to the right, is ask the question, where does a ligand bind, or epitope mapping, or conformational change of a protein in response to a drug perturbation? How is a complex assembled, et cetera, et cetera. And then, of course, we have a class of more challenging proteins that are disordered proteins, membrane proteins, and glycosylated proteins. So there are several advantages of using hydrogen deuterium exchange, and I append those here. So first of all, you need to have your protein in a native environment, which is great. You need low protein amount, microgram level amounts. The analysis itself is relatively rapid, and I'm going to show you in more detail in a few slides. You can probe dynamic regions that are often not present in high resolution structures that are available. And in theory, there is no size limit of the things that you can analyze. So you can analyze a complex 1 megadalton, 500 kT, et cetera, et cetera. Of course, you cannot get a high resolution structure from this technique. So we consider this to be orthogonal to NMR, X-ray, or cryo-EM techniques. Okay, so moving on, I'm going to give you like a few details on the technique itself so that everyone can follow later on the applications. Starting off, what exactly is it that we measure by mass spectrometry? So there are 3 types of hydrogens on a protein, and we measure the exchange on the amide backbones. So as you all know, proteins are very dynamic. They live in a closed state, but they would pretty often visit the open state. And while they are in this open state, this is an opportunity for deuterium, if you have diluted your protein with deuterium, for deuterium to change from amide to deuterium. Sorry, the exchange will occur from the proton to the deuterium. And this different in mass, hydrogen weighs 1 Dalton, the deuterium weighs 2 Dalton. So this uptake in mass we measure with high resolution mass spectrometry. So there is a rate of exchange, of course, depending on the amino acid, and this depends on the hydrogen bonding. So what we know really well is that non-hydrogen bonded or dynamic regions will pick up deuterium faster, whereas regions that are hydrogen bonded and the amides are least accessible, these will exchange more slowly. There are other factors, of course, affecting the exchange, such as the temperature, the pH, and whether your region is solvent accessible. But at the very end of the day, you can directly correlate this deuterium uptake to protein structure interactions and conformational changes that occur. So how do we conduct these experiments? The first step is, and I'm showing here for a very typical example, for instance, asking the question, where does my ligand bind and whether it binds? So you have your protein in native buffer, you dilute with deuterium 1 to 10 or 1 to 20 times, and then the protein will start deuterating. And then you have another sample where you have your ligand now present, and you will do exactly the same. So at the end of its time point, typically we start from few seconds, extending to a few hours. You want to quench the reaction, and we do this by dropping the pH to 2.5 and temperature at 0 degrees. At this stage, we add a very high concentration of guanidine hydrochloride because you really want to denature the protein at this point to be able to digest it very fast. If you have disulfides, we also add TSEP, and then we can move on to the analysis. So there are a couple of options on the analysis side, and the first one and the easiest one, the most simple one, is to actually measure your protein intact and ask the question, does my ligand bind? And this is very useful for ligand screening cases because you can have these results within a day. So you can learn a couple of things from such a graph. What I'm showing here on the y-axis is the number of deuteriums that you take over time, and the x-axis is the time. So you see already here in this example, at 10 seconds, 70 residues have exchanged from proton to deuterium. That means that already you know that in your protein, 70 amides are not participating in hydrogen bonding. And then, of course, if you compare the curves plus and minus ligand, and you see a change between the two, that's a positive indication that your ligand binds. Now, the next question is, where does it bind? And for this, we need to digest the protein. The technologies now that are out there and the proteases are really good in this. So what we do, we have an online column that has typically more than one enzymes that are non-specific. So we generate a plethora of ions of peptides that I'm showing here. These are overlapping, and we want to get full coverage when we do this. We analyze each sample after the digestion step that it's typically 2 minutes. We analyze each sample by LCMS, and then we try to figure out which peptides actually show a difference on the deuterium level upon ligand binding. So these are like more conventional workflows. We call them bottom-up HTX. And you can learn again about protein dynamics and interactions. And now you can localize where your ligand binds or where your epitope is, etc. And data analysis now, depending on how many states you want to probe, may take from a few days to a few months if your protein is large and you want to screen many states. However, we are very excited to have new tools in-house that allow analysis of this data within a day. And this is using DIA approaches that I'm not going to go into, but it's very exciting for us because we have spent days and days on the data analysis side. And the last thing that you can do is using computational approaches. You can even increase the resolution of your data. And that happens only if you have many, many overlapping peptides. But in this case, you can actually localize the deuterium at the amino acid residue level. And this is really great, especially in protein-protein interactions, because you can point to certain residues that participate in binding. And this, of course, is very time-consuming, and we don't do it as often, I have to say. Okay, now that we have seen how the workflow, how we conduct experiments, let's move on to how we do things in our platform. And for this, for HDX, we have a state-of-the-art autosampler that I'm showing here from Trajan, and we work very closely with the company over the past few years. The autosampler, as you see here, is very carefully temperature controlled across all stages of sample preparation, because deuterium labeling is very sensitive to temperature changes. So we need to be really careful when we conduct these experiments. And to not introduce any, artifacts. So these are not trivial methods, because you have 10 compartments that you need to optimize. We have developed workflows at the intact 10 peptide level, and we use state-of-the-art mass spectrometry. But basically, it's great because now you need only a few picomoles of your protein per injection. So we have even lowered the amount that we needed compared to other generation instruments. Applications that we have span protein-ligand interactions, protein-protein interactions, epigenetic complexes a lot, IDRs, more challenging systems as are membrane proteins and nucleosomes. And one thing that we are really excited, you have heard about the importance of PTMs now on proteins and all the workflows we have developed in the lab. So on the HDX side, we use cell digest for this work. So basically peptides that are denatured, and we ask the question first, how does a PTM affect the rate of exchange? Because then once you go and do an experiment of an unmodified protein versus a modified protein, to be able to say if there is a real effect, first you need to know whether this PTM affects the rates of exchange. So for this work, we do it at the proteome level. And the idea there is actually to use machine learning approaches in the future and AI-based models to revise the HXMS rates. And I have to say that depending on the project needs, there are also other structural approaches we use, mainly cross-linking mass spectrometry and native mass spectrometry, which is great for deducing protein ligand stoichiometry or higher order structure. So we have a nice selection of approaches depending on the question. So when projects come to us, we really need to do some mini method development because each protein is unique. And how do we start? You can really divide the HDX process into the labeling and the quench and digestion steps. So starting, we start working backwards actually. So if you come to us and ask for HDX, first we need to answer the question, is it really feasible? And do we get full coverage of your protein of interest? And this is a step where we need to optimize the quench conditions, whether to use guanidine or urea, how much of that, and then TSEP. We only have 2 minutes to denature the protein and reduce disulfide bonds. So we are talking about 2 molar, 3 molar guanidine or urea at this point. And then nowadays, there are many proteases that are commercially available that offer high sequence coverage and resolution. So this process takes about a day or two to be able to get back to the collaborators and say, okay, we see what we want to see, we are good to move forward. And then, of course, you need to ask, to set like the labeling temperature, right? At what temperature do you label? So in many cases, using room temperature will do for many of the proteins, because what you really want to do is within your sampling window that extends from 10 seconds to hours, you want to get like a full curve on peptides, starting from lower deuterium uptake to higher deuterium uptake. However, there are cases that I'm going to show in a couple of slides down in the presentation where you're interested both in slow exchanging regions and in fast exchanging regions. And if you want to compare now state A versus state B, it could be that within your sampling window, this exchange occurs either at later time points or at very fast time points that you are not able to probe. So what's the way to work around this is play with the temperature. So if you increase the temperature, you increase the dynamics, the kinetic energy of the protein. So the hope is that you will get this in your sampling window, this exchange. And then if you slow down, if you decrease the temperature, then you can also slow down the dynamics. So then you have a chance of actually moving this into your sampling window and seeing that again. So there have been projects in the lab where we needed actually to work all the way from 35 degrees Celsius down to 0 degrees Celsius and get a full picture of protein dynamics. Okay, moving on. What is needed for a successful HTX experiment? As I've already mentioned, you need a purified protein, complex, or even a co-IP samples in the case of drug screening. 50 to 100 micrograms is a good starting point. And I'm saying in small aliquots because people come to us and they have a milligram aliquot. So basically after we thaw it, we just discard it because we cannot use it for structural studies. So small aliquots are very much preferred and appreciated. Then we need the functional state of the protein. We need this protein to show that it is pure. And of course, sequence any tags, modifications that are there, any disulfides, etc., and information you have around this. Whether the protein is a monomer or a multimer also is something that we would like to know. And any additional, of course, biophysical or structural data are always helpful. And for applications as is ligand binding, and by ligand here, we can really probe small molecules, nucleotides, or peptides. A known KD always helps. And why is that? And I have this cartoon representation here to the right to explain how we set up these experiments a little bit. So if you mix the protein to the ligand 1 to 3, assuming that your ligand is soluble, if you have a strong KD 10 nanomolar, what you see here before labeling, then all of your protein molecules will be bound to the ligand. Then, however, you dilute your protein with deuterium, and if your KD is small, that's great because still 100% of your protein molecules will be bound to the ligand. And you really want a high percentage bound because then data analysis becomes really complicated if not most of your protein molecules are bound to the ligand. You measure the average of all protein molecules. Now, then if you start working in the micromolar range, you see that even before labeling, you may have a high number. However, once you start diluting, these numbers drop significantly. So for us, it's very important to know roughly what the KD is to avoid the scenario where we work in the range of 50% of your protein molecules being bound to the ligand, and then you try to figure out what the result is and where does the ligand bind. So this was pretty much an introduction to HTX and how it works and the workflows that we have established. And over the last 4 or 5 slides, I want to show you specific examples from collaborations we have had in the past. And actually, I want to start with a project we started working a month ago that has to do with protein low affinity ligand interactions and see how low can we actually go. And unfortunately, I cannot reveal anything about the project, the protein, or the ligand itself, but I want to show you how, in this case, we set up experiments to get meaningful information out of it. So when the collaborator approached us, they told us that they had done a virtual screen using billions of small molecules, and they were able to nominate a few small molecules binding to their protein of interest, which is a multimer. So the question that they first asked us to say, can you detect that the ligand is even binding so that we can confirm what the virtual screen has said? So this is where, again, intact protein screening comes in very handy because you don't need to lose the time to go at the peptide digest level unless you have indications that your ligand binds to the molecule, to the protein. So the experimental setup in this case, we used 10 times the ligand concentration that the protein, and we were lucky because the ligand didn't behave weirdly in DLC, did not precipitate, so we were still able to get really meaningful data. When you work with ligands that are that high and they have such a low affinity, you really need to do a small dilution with deuterium. You cannot do a 1 to 20, that is very typical in the field. And one extra step that we do, we actually spike the ligand in D2O. So then once you dilute your protein into D2O, the ligand concentration remains the same, even though now you have diluted your protein concentration. So this only helps. And to show you numbers, if you conduct the experiment in this way, actually the KD for a first ligand we had was 8 micromolar, and during labeling, we were able to have 92% of the protein molecules bound to the ligand. And then for a 20 micromolar ligand, we were able to get 85% bound, which is pretty great. And as you see here, the two curves, minus ligand plus ligand, actually in this case, the ligand leads to higher deuterium uptake, but this is going towards the correct direction because the mechanism of action of these ligands, they are supposed to disrupt the multimer to a monomer. So now you have more amides exposed, and then in the presence of the ligand, you get higher deuterium uptake. And another thing I want to mention, that especially when you do small molecule analysis, you get very small changes, as you see here. So in order to get statistical significance, you really need to do many replicates so that you reduce these error bars. And what we are doing now, of course, on this is we do HTX at the peptide level, because the next question is where do the ligands bind? But I'm showing all this just to say that even if you have a ligand that has like a not so great affinity and you're down 20 micromolar, there is still a way to do it. Okay, moving on now to a more traditional example, I should say, is how do we use HTX-MS to probe protein-protein interaction interfaces? And this is the case of PD3A and SLAFN12 that we have published actually now for a few years. And I won't dive into the biology because I just want to show you from a technological standpoint how we conduct these experiments and why was it important to use HTX in the entire process. But if you want more information, definitely check out this paper by Colin Garvey and Zhou Yun and Heidi Grulich. So that was a project that was cooking for a few years, and they were able to see that a subset of these PD3A inhibitors kill selected cancer cells. And doing proteomic studies with our group, what they were able to show is that SLAFN12, actually another protein, is required for this cytotoxicity. So they were able to get X-ray crystals of PD3A, showing here that this is a nice homodimer. However, when they tried to put the entire complex together, they were very unsuccessful. And they were doing site-directed mutagenesis at the same point, but they needed a fast answer of figuring out where is the interaction interface of PD3A with SLAFN12. And this is what we did. We performed an experiment where we used two states. We compared the states. One was PD3A with TP51, and the other one was PD3A TP51 in the presence of SLAFN12. And the KDs between PD3A and SLAFN12 in this case was low enough to allow for one-to-one mixing of the two proteins. So during labeling, we had 87% of protein molecules bound to SLAFN12, which was great. And what I'm showing here to the right, that I'm not sure that you see because it's very faint, but this is the sequence of PD3A, and we get hundreds of peptides. And then we ask the question, what peptides show a decrease in the deuterium uptake in the presence of SLAFN12? And I'm having here an example to the right where you see the two states. We got many peptides showing a decrease in deuterium uptake, and this suggests the potential interaction interface between PD3A and SLAFN12. And I hope you can appreciate that when you work with protein-protein interactions, the changes that you see are way bigger than what you get when you work with small molecules, because you have more amides basically participating on the interface here. So we were able to get three regions that you see here in green, where all overlapping peptides show the decrease in deuterium. And if you spatially map this in the PD3A structure that was available back then, we see these three regions. One is on the dimer interface. So basically presence of SLAFN12 makes the dimer more compact. And then we got another region down here at the bottom of the protein where it showed to be protected. And eventually we solved the cryo-EM, showing beautiful alignment of the HTX data with the cryo-EM, because they were able to show that the SLAFN12 dimer actually binds to the PD3A dimer, and the regions here overlay. So that's a very typical example of how to obtain information on an interaction interface between two proteins. Moving on now to another challenging application that we were working for I think 2 years, and Amanda actually, who's here today, that was her entire PhD project. So I have only a couple of slides just to show you because this was like a very tricky one. So Amanda was working, she's in Brian Liao's, she was in Brian Liao's lab as a PhD student, and she was working on LSD1. LSD1 is a fat-dependent H3K4 demethylase, and it's part of this LSD1 Corest H-tag complex, and is recruited by transcription factors in these genome binding sites to repress transcription. So the crystal structure of LSD1 in the presence of Corest was solved back in 2006. And as I'm showing here at the bottom, this has an intrinsically disordered N-terminus that, of course, was not captured in any of the high-resolution structures that are available, followed by well-structured domains, the Deuterium domain and the amino oxidase domain, that's separated by this protruding tower domain. So Brian's group has developed this CRISPR suppressor scanning, as he has termed, where they introduce all these mutations across the entire length of LSD1, and then they treat the cells with the drugs, and they are able, at the end of the day, to identify resistant mutations upon these drug perturbations. And they have come up with a score, and what it's seen here, and this is actually a paper back from 2019, I encourage you to have a look at it. Many of the mutations that light up, they are in the active site as expected. But what was not expected is that many of the mutations actually cluster at the N-terminus of the protein. So they were not sure what's going on, but as you see, not only for one drug, they had seen the same thing for 2 more drugs. So what is happening, and how can we actually understand the role of the IDR in protein structure function? So Amanda was able to expand cell lines harboring deletions of amino acids, of hydrophobic amino acids at the N-terminus. And for this experiment, what we did, we compared the wild type versus a mutant one, that this is a 10 amino acid mutation at the IDR, another 10 amino acid further down the IDR, and then a mutant that the entire IDR is missing. And we ask now the question, what can we learn in terms of structure for these mutants, and why the different phenotypes? So I'm going to summarize just in one slide that doesn't serve justice, thinking that this was like a project that was happening for 2 years. However, we got some really useful information that there was no other way of getting. First, what we saw is that all peptides in the IDR, they are in an equilibrium between a closed and an open state. So what happens is actually IDR is getting deuterated really, really fast. So by 10 seconds, you have already reached your full deuteration levels. So we had to do the experiment at 0 degrees and then do very early time points manually in this case. And you're seeing that instead of getting a Gaussian distribution, you get 2 populations below this distribution. So that indicates that you have a closed and an open conformation, and then the tail interconverges between those 2. And that was the case for all peptides in the IDR region. However, when you introduce the mutations, you see now that the fraction of the open state is higher in N delta 1, and in N delta 2, it's almost fully open. So basically what we have been able to show is that the mutants favor a more open state of the IDR. And then zooming out of the IDR and looking at results all across the protein, we were able to get a number of regions that showed to be more exposed in the mutants. So all these regions that we captured, these are potential interaction interfaces of the IDR, transient interactions with the structure core, and we were really happy to see that. And then eventually we came up with a model where wild type is favored in the closed state, where it's less accessible, but you can disrupt these interactions here by either mutations on the IDR or the structure core, and then favor an open state that now is more accessible, and you have a different transcription factor interactomics landscape. And there is really no other way because it's an IDR, so there is no really high resolution technique that you can use for that. And having said that, I want to show an example here of AlphaFold, right? We live in the AlphaFold world. So why is HDXMS valuable? Literally, you can type in the name of the protein these days and get a structure, a model of from AlphaFold. However, what's really important is this model confidence graphs here at the bottom, where if these are dark green, it means that the model has very high confidence and the amino acids are positioned really nicely. But what is shown here in lighter green is that the confidence is very low. And this is what AlphaFold shows you for LSD1, but if you compare it to the image I showed you earlier, this does not agree with what we've seen from HTX experiments. And especially when it comes to transcription factors, 50% here of the protein is very low confidence, and here even like 90% of the protein is of very low confidence. So we really truly believe that HTX-MS can shine in such cases where you are interested between protein interactions that are mostly disordered, and you are able to capture how do these interact. Okay, and this is my last slide. Over the years, we had an interest from many BroadE members about doing HTX for membrane proteins and characterize the membrane protein dynamics, because as you know, membrane proteins are tricky to get like a cryo-EM or an X-ray structure, and you need to develop methods for that. So there are a couple of options here. You can reconstitute membrane proteins in detergents, and two favorable detergents for the mass spectrometer are these two that I append here. These are non-ionic and they don't interfere with the analysis. Or alternatively, you can put your protein into a nanodisk where you use the natural phospholipids, and then you can have a membrane scaffolding protein keeping the whole structure intact. And actually, a rotosampler that I showed you earlier, we have an online, it has one of its capabilities is to remove the lipids online following the labeling and quench. So that is great because these don't interfere with the analysis on the mass spec side. And what I'm showing here is some first results from aquaporin Z that we use only for method development purposes here. Aquaporin Z is a homotetrameric transmembrane protein that moves water molecules across the membrane. And it's a very difficult one because as many membrane proteins, it's extremely stable and it's very difficult to digest. So if you see actually up here, not much exceeds outside the transmembrane region, right? So it's very difficult to denature it and access it with digestion proteases and actually get a coverage map. So this is how, though, the peptide, after optimizing such a method, we were able to get 90% coverage of the protein with many overlapping peptides. And when we did a first round of HTX experiments extending up to an hour, what you see here that whatever is embedded in the detergent in this case gets very low deuterium uptake. So it's a very small protein. So in such a case, we'll need to increase the temperature and extend the time point so that we are able to allow time for the protein to move and get deuterium dynamics. And where do we want to go with this? This is very preliminary, and the postdoc in our lab is working with this, but we are very excited to collaborate with Mahmoud Nasra at Harvard University, who is also here, who they have this unique technology in their lab of preparing these covalently circularized nanodiscs of many of different diameters. And the goal there is to use different diameter nanodiscs and actually not only be able to fit one molecule of your membrane protein, but also two copies and actually start building larger complexes and analyze those by HTX-MS. And that will include ion channels, GPCRs, and in general, larger protein assemblies. And that was my last slide, and I hope I have been able to give you a flavor of what we can do with this technique. We are happy to hear from you if you have challenging systems or not. And I would like, of course, to thank, we are very consistent in using the same photo list. Yeah, we need a new one. The BroadE Proteomics team and our collaborators over the years who have helped us like develop the methods and get like really meaningful results. Thank you.
Awesome.
Oh, okay.
So you mentioned that some of those analyses took you 2 years to actually finish, but there's been a lot of improvement in the software and automation of it for being able to do the analysis. Have you thought about going back and taking the data, running it through the new pipelines that you have to see whether you can fundamentally replicate the findings that took 2 years now in the space of a week, for example?
That's an excellent question, and I have thought about it. We would need to reacquire everything in the DIA mode to do that, but yes, it's in the plan, of course, yes.
And one follow-up, at the aquaporin, how many passes? A single pass, multi-pass, do you know? I think it was here.
I think it's like 4 or 5 passes. Yes, but it's like a tetramer, as you see here, so that this is a tetramer there, but yeah.
To increase the coverage of transmembrane domains?
Very good question. So we tried 5 different, if not 8 different quench conditions. So we used guanidine, we used urea, we used the protein was crashing. You know, yes, but the protein was crashing. We didn't like that at all. What we ended up doing, actually using only formic acid, and then use specific proteases, non-specific proteases, sorry, nepenthesin is a protease from plants that is very non-specific and digests pretty much everything in combination with pepsin. So we used this dual protease column that I think actually led to, yeah, coverage of those, because using... No, at this point, I think it's 5% formic or less. Yeah. Yeah. Yes.
I was wondering if instead of using G2O,
I'm sorry, I can't hear you.
instead of using G2O, Deuterium labeling, can you use something like an amine reactive agent like an NHS ester or something?
So not for this kind of work. I'm sure you can do it in other cases, especially if you want to go over proteome level samples. But in this case, the time that the labeling occurs is very significant. We need something that labels from 10 seconds to a few hours so that we can very accurately control that. So for this particular technique, it's only deuterium. And you also don't want to introduce something that you will perturb the system, right? Because this is just a different isotope and this is what we would prefer. So yeah, so you don't get any artifacts. Okay.
All right. So for my talk, I'm going to give you a high-level overview of what we can do with affinity proteomics, proximity labeling, and target identification of small molecules. So I think, Steve nicely introduced some of these concepts already, so I'll just quickly go through them. Affinity proteomics it's a common workflow we use in proteomics, and it's often hypothesis guided. You have a protein, you're really interested in what it's binding to in your system of interest. So we can do experiments to look at protein-protein interactions, protein nucleic acid interactions, or protein small molecule interactions. And these types of experiments can help you answer questions like, what does my protein interact with, or what does knockdown or overexpression of my protein do, and how does it change the interactome, or how does a mutated version of my protein change its interactome? So the way we do these experiments is, of course, to start with picking your bait protein of interest, and then you need an antibody to enrich that protein. You can do this one of 2 ways. Typically, you want to start with an antibody to the endogenous version of the protein if you can, because that's great, because you don't have to add a tag and change anything about the protein. But that's not always easy. Sometimes there's just not any good antibodies for your protein, or proteins are really low level and the enrichment is not good enough. When that occurs, people often use affinity tag constructs of their protein. So small tags, flag tag, V5 tag, those work really well. Of course, you have to play around with the expression level. You don't want to slam it and have extremely high expression and create artifact interactors. And then there are nicely built-in universal controls like empty vectors. And this is also a way to combat low expression of proteins. So we can use affinity proteomics to identify interactors of a protein of interest, but we can also use it to identify drug-dependent protein interactions. And so I'm showing you an example from an older paper. It's an oldie, but I promise you a goodie. It was some of the early work in trying to understand the mechanism of action of a molecular glue that's used to treat multiple myeloma, lenalidomide. And so in this experiment, the first step was to try and identify the interactome of an E3 ligase CRBN. And so we pulled down CRBN, and what we found was the ligase components, Col4A, DDB1. But then we also pulled down CRBN in the presence of the drug lenalidomide. And what was so beautiful to see was that not only did we pull down the E3 ligase complex, but we also found that this drug increases the interaction of two very important transcription factors in multiple myeloma, IKZF1 and IKZF3. And in other work that went into this study, we showed that the drug increases the binding of these transcription factor proteins to the ligase and promotes its ubiquitination and degradation. So this is just a really nice example of how we can look at not only protein interactions, but those that depend on drug. So on the data analysis side, there are a lot of web-based databases that contain information about protein interaction networks. And I just wanted to spend one slide talking about this because they're really valuable. A lot of you may be familiar with the string database. It's really easy to use. It contains a lot of information. So string contains interaction networks based on experimental data, but also computationally predicted data. So this comes from text mining, genomic features, orthology from other organisms. So for this example, I put in CRBN, and maybe a little bit tiny, but you can see that the network contains Col4A, DDB1, and also these transcription factors, IKZF1 and IKZF3. String also has functional enrichment information. So it pulls in GO, PFAM, KEGG pathway information, and it's really easy to use. You get a score for these interactors in the network. And the edges here, the thickness of the edge is usually related to confidence, and then the nodes are different putative interactors. I also wanted to point out another database that's been generated across the river in Steve Gigi's lab at Harvard Medical School. So this is called Bioplex. This network database is a mass spec-based experimentally derived protein interaction. It's made of experimentally derived protein interaction data from mass spec experiments. So what they've done is to tag up to 10,000 baits, and so they have up to over 100,000 interactions. And so you can see here, I put in CRBN, and you get a network here, and you do get Col4A, but you don't get IKZF1 or IKZF3, and that's because context matters. So they've done this work in a limited number of cell lines, of course. So this is 293 T cells and HCT116. So these interactions may not be occurring in these cell lines. So the work we had originally done were in multiple myeloma cells. But this is a really nice tool to say what has already been seen by proteomics and mass spec. All right, so moving on, I want to talk about proximity labeling that you heard a little bit about from Steve. So proximity labeling, the goal is you can do interaction mapping, but it's often used for spatial mapping in living cells. So why do we need proximity labeling? So we all know that we can fractionate different regions of the cell. However, there are compartments and complexes in the cell that can't be purified easily. When you lyse cells, you disrupt the native cell condition. Of course, there's immunohistochemistry, but that's always dependent on the quality and availability of an antibody, and often this can be low throughput. Also based on antibodies is, of course, fluorescent imaging, but you don't always have the resolution to ascertain subcompartmental localization. And then again, it can be fairly low throughput, although this is improving these days. So proximity labeling. Proximity labeling is a really valuable tool coupled to mass spectrometry for studying proteins in living cells with high spatial and temporal resolution. So it's really a complementary approach to some of these methods for interaction and spatial composition mapping. The enzymes that we're typically using for proximity labeling were developed by Alice Ting, who you heard from Steve. She was at MIT and now she's over at Stanford. So we've worked with Alice's lab now for over 10 years to couple proximity labeling to proteomics and mass spec to do spatial mapping. So Alice's lab has developed the two enzymes I'm showing you here on the slide. Apex peroxidase is the first enzyme. So the way this works is that you have these ligases. They're tagged on a protein of interest that's going to a region of the cell of interest. And Apex catalyzes the oxidation of a biotin, in this case it's biotin phenol. It's a short-lived biotin phenoxyl radical in the presence of hydrogen peroxide onto tyrosines of proximate proteins within nanometer distance. She's also developed TurboID, which is a complementary ligase. This ligase was evolved from BroadE, so BioID, which you may be familiar with. This uses ATP to convert biotin into a reactive biotin species, and then this biotinylates lysines on proximate proteins. So this works quite well for spatial mapping. These enzymes have different characteristics, and they're also complementary. So I just told you they label different amino acids on proteins. That's one thing, but their kinetics are also different. So Apex is very fast. In about a minute, you get labeling. TurboID, you're going 10 minutes to an hour. So it really depends on the question. So this is the typical workflow we use for proximity labeling. You usually, as I said, you have your proximity labeling enzyme, and then it's conjugated to your bait protein of interest that's in your location of interest in the cell. You also, in these experiments, have to be careful about including the right negative controls, and these negative controls include a spatial control. And why is this? Because proximity labeling is not happening at a fixed radius. It is labeling promiscuously and proximate. So you can think of it as a contour map where the labeling falls off nanometer by nanometer from the source. So if you're trying to map proteins that are in unbound compartments or face the cytosol, you need a spatial control, right? So that's quite important. And so typically we're comparing samples that are labeling our protein of interest. You have your spatial controls. Because you've modified your proteins with biotin after the proximity labeling reaction, we can use streptavidin, pull down the proteins, do on-beat digestion, use TMT as you've heard about, and look at the relative quant to identify your proteome of interest. So typically in these experiments, we're identifying proteins, but we can also identify the sites of biotinylation in a complementary way. So a few years ago, we developed an approach using anti-biotin antibodies to actually not enrich the protein, but enrich the biotinylated peptides, just like we do for phospho, as you heard, or other modifications. And this can be really powerful if you want to understand protein topology, for example. So is my protein exposed on this side of the membrane or the other? So over the years, we've used proximity labeling for a number of applications. Our earliest application was in collaboration with Alice and Vamsi Mutha's lab. And we use proximity labeling to map the proteome of the mitochondrial matrix as well as the proteome of the intermembrane space. This is just a list of some of the papers that you can look up. And I'm happy to talk to you more about different applications, but we've also used proximity labeling to map the proteome of the synaptic cleft in stimulated and unstimulated conditions. We've used proximity labeling to map contact sites between ER and mitochondria. We've also used proximity labeling to map the cell surface proteome of different cells in fly brain. So there's really a lot of different applications for this technology, and it works quite well both in cells and in situ. So I wanted to just spend a few slides telling you about some of the most recent work, and it's a really nice and exciting application of proximity labeling. And this was done in Alice's lab, and the work was driven by a really talented student in Alice's lab, Jenn. So this approach is called Transit ID, and the approach measures trafficking of proteins in the cell. So this builds upon proximity labeling and uses dual enzymes to map how a protein moves from point A to point B. So it's a little complicated. I'll just walk you through. So you have a cell, and now you're expressing both proximity labeling enzymes I showed you a few slides ago, TurboID and Apex. So TurboID in this case is expressed in a source location, and Apex is expressed in a destination location. You do the TurboID proximity labeling reaction, and then you have a chase period, and that can be minutes, it can be days, whatever the question you're trying to answer. And then you do a second reaction with Apex, and now you have proteins that have gone from point A to point B. If they've come from point A, they should have two different labels on them. You do a dual enrichment, and then we analyze the samples by mass spec. And so we've applied this to understand how proteins form stress granules under stress. In this example, we use transit ID coupled to TMT to resolve the spatial origin of nuclear encoded mitochondrial proteins. And so most mitochondrial proteins, as you know, are translated in the cytosol, but there has been work that suggests that a subset of these proteins are translated more locally near the OMM to facilitate co-translational imports. So we asked the question, can we use transit ID to identify those proteins? And so we had cells that expressed turbo ID at the OMM and apex in the mitochondria, or cells that express turbo ID in the cytosol and apex in the mitochondria. And then what we do, we use TMT, so we compare the extent of protein enrichment intensity to then calculate which proteins are more local to the OMM. And so by doing that, we were able to identify 148 proteins that were candidate mitochondrial proteins translated at the OMM. To validate those findings, Alice's lab went on to label cells with a molecule called OPP, opropargyl puromycin, that labels nascent proteins. And so labeling proteins with OPP followed by a single proximity labeling step, they were able to show that indeed, I know this is a complicated plot, but if you focus here, indeed, proteins that we identified through transit ID and proteomics were enriched and as nascent proteins at the OMM. So that was really nice to see that transit ID could be used for this application and validated. Okay, so moving on to cell surface proteomics. So cell surface proteomics is a really important tool. There are a lot of very important molecules on the cell surface that are drug targets. So this is a common application of proteomics. So the sort of general conventional way to do cell surface proteomics is to use a molecule like this, biotinylation, and you biotinylate proteins on the cell surface. It's an NHS ester, and you biotinylate lysines. This is a cleavable molecule, so you can eventually release the protein and identify it. You can do this sort of work, not just in cell lines, but it can also be applied to human-derived clinical samples. So in work that is submitted, our collaborator, Jifu Lee, who is at Genelia, isolated blood from healthy donors, and on the same day isolated dendritic cells. Cells were stimulated with TLR2 or TLR1 ligand, and then we did cell surface proteomics to identify those proteins or how the surface of dendritic cells was changing under stimulation. So that's just an example that you can do this work not only in cell lines, but in clinical samples. And I told you, and you heard earlier from Steve, that you can also use proximity labeling to do cell surface work. And it's a really nice application of proximity labeling because you can do this work in situ. The way this works is to use a proximity labeling enzyme that's actually active in the extracellular space, so that's HRP, and then you use a biotin probe that's membrane impermeable, so that's important. And as I said, we've used this technology to map the synaptic cleftome. We've also looked at the surface of proteins on the surface in both fly brain as well as in murine samples. And we've looked at cell type specific temporal resolution upon aging or drug treatment. All right, now the last thing I want to talk about is target ID. So the question is, if we have a small molecule, how do we identify its protein targets? So the classical way to do this is to take your small molecule, tether it to an affinity matrix or a bead, and go fishing in cell lysate and identify which proteins are binding to your small molecule of interest. That works, and it has worked. That workflow is outlined here. So as I said, you have a bead with your small molecule, you go fishing in cell lysate, and then you use a soluble free competitor of that small molecule to identify which proteins are specifically interacting with your small molecule. Xiao and Ong and others in the group years ago have used this to study targets of the staurosporin analog K252A, and that worked really well. Staurosporin is a high affinity, it binds a lot of kinases, and that worked quite well. However, there are disadvantages to approach like that. and I think as Steve mentioned, not all compounds will retain their activity when you chemically modify them to link them to an affinity matrix or a bead. So to figure that out, you need extensive chemistry support and SAR development, which you don't always have. And of course, this lacks observed compound activity in a cellular context. So what would be transformative to us? Can we do this without modifying the compound in a cell or in a cell extract? And so for the rest of the talk, I want to focus on thermal profiling, which does not require modification of the compound. So there are different flavors of thermal profiling that I'm going to talk about. We have CETSA, PPP, and PISA, but essentially they all work in a similar way where you are treating with a compound and then doing a heat treatment and tracking the change in abundance. So I think you saw this graphic earlier in the session, but just to remind you, these thermal shift assays work because when a protein is subject to heating, it starts to unfold and eventually aggregates. And so as a protein starts to unfold, this is called its melting temperature, and that temperature increases when it's bound to a ligand. So this has been used obviously in pharma for a very long time and measured out by fluorescence. And then 10 years ago, it was discovered that you could do this in the cellular context. So you could do this heat treatment in cellular extracts and then go blot, in this case, Western blot for your protein of interest to see, is its melting curve changing when it binds to a drug, and so making it a candidate for your drug target. But that's slow. It's one protein at a time, and you have to know the protein you're looking for, right? So how can we do this in an unbiased way? So in 2014, Savitsky et al. Put out a very nice study where they showed that you could couple thermal profiling to mass spectrometry for unbiased target ID. So I'll just walk you through how that works. So you either start with live cells or cell extracts, and you treat with the ligand. And then you subject your samples to temperature treatment. So this is about 10 to 12 different temperatures. And your proteins will start to aggregate at higher temperatures. You remove those aggregates, and then you measure the soluble samples, and you measure all of them, and you look to see which proteins have stabilized versus in ligand treated versus non-ligand treated cells. As you can imagine, I told you there's 10 to 12 temperatures, there's plus drug, there's minus drug. That's a lot of measurement time and a lot of samples. So PISA, as you heard about earlier, is a higher throughput version of thermal proteome profiling that instead of measuring each temperature point individually for treated and non-treated samples, we combine all of those temperature treatments for ligand treated sample and all of the temperature treatments for non-ligand treated sample together. And then what you're doing is measuring this area under the curve. So really you have two samples, right? You have treated and untreated, and both have seen a temperature treatment. So this really reduces mass spec time and sample consumption. And so the output of a PISA experiment is not a curve now, so you're not going to identify the melting point, but simply a ratio where you're looking to see which proteins have been stabilized or increased abundance in the presence of drug. So some things to think about with PISA. Do you start with cell extracts or do you start with intact cells? Because you can do both. So it depends. So the general most common way is cell extracts because you want to get at direct targets, right? But the downside is that proteins, metabolites, cofactors, they'll be in a diluted state and normal metabolism of the cell is stopped. So if your drug depends on metabolism or cofactors, that could create a complication. But this is usually where we start. You can also do this in intact cells where cellular machinery is active. So that's great. And it is possible to observe the direct targets, but most likely, depending on the concentration and the time, you're also going to get the secondary targets. So if the drug stabilizes, inactivates, or activates your primary target, you may see the downstream effects, and then it's hard to disentangle what's a primary target, what's a secondary target. The other thing to think about in a PISA experiment is what concentration of my drug do I treat with, and is what's the temperature range I should use? Because if you go too wide, it may dilute the effects. So these are questions we have to deal with on a case-by-case basis. But factors that influence the drug treatment concentration are, of course, the IC50, the solubility of the compound, the binding affinity to any known protein targets, and any previous experiments in the literature. Typically, we start high because we want to make sure we see something, and then we can titrate down from there. In terms of the temperature range, we're usually going from less than 44 to around 60 something. That's a general range, but this can also be scaled. There are databases on Proteomics DB, I put the website here, of meltome curves for proteins. And so if a putative target or a known target is known, we can go into those databases and see what is the melting temperature and make sure that that temperature is in the range that we're going to profile. So this is the typical PISA workflow. So as I said, you can start with cells or cell extracts. We do this in a TMT 18-plex format. So oftentimes we'll treat with multiple concentrations of a drug to make sure that it's high enough to actually see a target, but also we can sometimes see a dose-dependent change, and that can be really helpful for determining specific versus unspecific hits. The samples are aliquoted, it's put into a 96-well plate, you do the heat treatment, and the critical step here is the centrifugation. So after the heat treatment, you want to remove the proteins that have aggregated and then only move forward with the soluble fraction. You recombine the samples, all for each sample, you recombine the temperature treatments into one sample, and then we move on with our typical digestion TMT workflow, fractionation and measurement. So one of our earliest examples was going back to staurosporin. I showed you that was successfully done by the classical target ID method. So does PISA work to identify the targets of staurosporin? And indeed, it worked beautifully. So we got high depth of coverage. When we treated cells with 1 micromolar staurosporin in the PISA experiment, we identified over 8,800 proteins and over 300 kinases. And this line shows you all the blue dots above the line are kinases that were significantly stabilized in the presence of the drug. So we identified 17 known staurosporin kinase targets, so that worked quite well. And because this works so well, we now include staurosporin as a control arm in every PISA experiment we do. And PISA, I think, is a really nice complement to some of the other discovery workflows that you've heard about earlier today. And I'm going back to this older study, but it's really valuable, and I'll show you why. So in this study, we did global proteome and ubiquitin profiling to identify, to try and understand the mechanism of action of this very important drug, lenalidomide. So you treat the cells with lenalidomide, and you see a reduced level of these important transcription factors, IKZF1 and IKZF3. And as I told you earlier, it's because it's promoting their ubiquitination and degradation. However, you don't pull out what the drug is targeting, which is CRBN. That doesn't change here because it's a molecular glue. So now if you do PISA and you treat cells with lenalidomide, you get as one of your top hits CRBN. So I think this really paints the full picture and is complementary for identifying the target of molecular glues. All right, so that's what I have. So in summary, I hope I've shown you that affinity proteomics is a simple but really powerful way to get at function and biological understanding of your protein of interest. And proximity labeling is a method for spatially profiling proteomes as well as surface proteomes and interaction landscapes. And finally, target ID can be done high throughput and in an unbiased manner without having to modify your compound using thermal profiling by PISA. And the outlook here, and you've heard a lot about improving throughput using this DIA method, we're also coupling that now to PISA. And what I didn't show you was important work done by Keith Rivera and Isabel Cressy in our lab, where they have now started doing all of the lab pipetting on robots. So it's making it faster, more reproducible. And we aim to couple that to high throughput DIA and really build out, hopefully, a really nice screening method for compounds. And so it's Groundhog's Day. You see this picture again. Thank, of course, the entire proteomics group, our funding, and our longtime collaborators, Alice Ting, who led a lot of the work that I showed you, and Ji Fu Lee. Thank you.
Questions? Cora, what would be the required proteins input for the pizza workflow?
Yeah, it's not small. So it depends on how many concentrations you want to do, but I think we're looking at a couple hundred micrograms right now, but I'm certain that's going to go down. Mhm. Mhm.
So small molecules can also destabilize protein.
Absolutely. Yes, yes, we have. That's a great question. So there are small molecules that can destabilize interactions as well. And so typically when you're doing these thermal profiling experiments, you're focused on stabilization, but we get everything here, right? So we get that information for free as well. So we can look to see what's been destabilized. And we've seen a few drugs that have done that. Mm-hmm.
And as a reward for hanging in there for the last talk, I will show you that brilliant proteomics group picture again at the end of this presentation. So like Steve mentioned, this is the data analysis overview of all the proteomics data that's been generated by all the previous speakers. I'm obviously Dr. Mani, I'm not Natalie Clark, as you can see. So here, my goal is to kind of present an overview of the tools and methods we use in the group. I think unlike previous BroadEs, this one I think will be more focused on what the tools are and how we apply them, as opposed to going into the details of the methods of the tools, the algorithms that the tools employ. For some of the tools, we are going to give like a superficial overview of how the tools work and what algorithms they use. But that is not the goal of this presentation. If you want more details about that, maybe you can visit me on the 5th floor or whatever. So Steve showed this slide in the beginning of the talk. And between then and now, we have added quite a few methods here, as you can see. No, I'm just kidding. This is a more updated version of the slide that Steve had. And I think the point of this is to kind of give you a collection of the types of tools that we use and where we apply them, right? So Steve talked about SpectroMill for analysis of raw data. So we have other, and many of the other speakers mentioned DIA data, which is a newer mode of collecting data on a mass spectrometer. SpectroMill does not analyze DIA data. So we have other tools for analyzing DIA data. And one of the things that we have done as we go along is to make more and more of our pipelines cloud pipelines. So the advantage is you can get effectively unlimited resources for the amount of time you need it, and then you're done. So we don't have to maintain large machines. And many of the newer projects have a lot more samples and a lot more data. So Claudia and others mentioned the Bruker machine, which generates exquisitely sensitive mass spec data, but they also generate significantly more data than the other machines in terms of just gigabytes of volume. And so all those can be much better handled by moving our pipelines to the cloud. So I'll go a little bit more into details about that when we get into Panoply, where Panoply is completely on the cloud. It's an automated cloud pipeline created primarily for the analysis of cancer proteogenomics data, like the data that Shanka presented. But it also has specific modules that can be more generally used. And the pipeline itself is not specific to cancer. So you can use it for other types of diseases also. So I will cover that. Prodigy is another toolkit that we have, which is a kind of the first go-to for people in the, for wet lab scientists in the group. So they generate some data, and the first level of analysis is either SpectroMill or one of the DIA analysis pipelines. And that will generate a table that you can then load into Prodigy and then start doing things like, how many missing values do I have? How many proteins did I quantify? If I have a simple wild type versus knockout comparison, can I do like a t-test to find what proteins are significantly different? Can I get a volcano plot? And in the proteins that are above the significance line on a volcano plot, what proteins are they? Do they belong to specific networks or not? Can I do like a PCA plot to see if my wild type and knockout samples separate or not? So those kind of basic analyses you can do with Prodigy. And then the other thing that, so BAM query is for immunopeptidomics data that Mavi already covered. And Quasar is for targeted proteomics data, which we are not covering in this session. So I think Steve mentioned targeted proteomics data, and we have customized tools for working with those. But the other thing that Natalie will cover is what we call computational consulting. So Namrata presented a lot of exquisite experiments where you're doing pull downs with a small number of replicates for a couple of different conditions and so forth. So in those situations, how do you find out whether something is differentially expressed or not? So many times you can't do like a simple t-test because she mentioned that there are all these enzymes that have to be there. So you do a series of negative controls with and without enzymes and spatial references and so on and so forth. So you need to take all those controls into account in trying to figure out what is really differential between the conditions of interest for you. So there we use a series of linear models ranging from a t-test is a simple linear model, but we also have a tool where you can look at more complex linear models. And many times the linear models are so complex that you cannot really automate it easily. So in those cases, people actually come to our offices with slides and say, this is our experimental design. Can you analyze the data or build a linear model for that? So we do that also. And I think in the computational consulting thing that Natalie will present, we are also trying constantly working with people in the wet lab to develop data for new data modalities, right? So you have PISA. It's a new method that people are working in the lab. So you get data from a PISA experiment. How exactly do you analyze that? What kind of models do you need? And what kind of approaches can you do to maximize the results you get from that? So that's kind of the overview of what the group does. I'll go into a couple of the raw data pipelines and Panoply, and then Natalie will cover Prodigy and what we do under consulting. So raw data processing basically starts with the output of a mass spectrometer, which is a binary file that can be anywhere from a few hundred megabytes to a couple of gigabytes. And from that, we want to get a table where for every sample that you have profiled, you have a measurement for either a protein or a phosphopeptide or some kind of what feature? Sorry, yeah. Some kind of a feature or some kind of a molecule that you're trying to quantify, right? So once you get this table, then you can use any standard data analysis tool, and Prodigy is one such tool that you can use, which has many specialized tools for the kind of data we get in the lab. But for raw data analysis, there are three basic steps. I think Steve went through this. I'm trying to kind of make it, repeat it and make it a little more concrete. So there are three steps. One is identification. The second one is validation. And the third one is quantification. So first off, you have to identify a peptide. And so for that, Steve mentioned we use database search. Secondly, once you have identified a peptide, you need to know how confident am I in this peptide that I have identified. Is it likely to be wrong, or am I quite sure that this is the correct peptide? So that is validation, and that's where you get calculation of false discovery rates and things like that. And finally, once you have a peptide, then you quantify the peptide. In other words, you say it was the measure was X in sample 1 and it was Y in sample 2. So that is at the level of peptide quantification. Steve also mentioned the complexities of getting to protein quantification starting from peptide quantification. The mass spec only measures peptides. So if you want quantification at the protein level, you have to somehow combine all the peptides you have seen for a protein and come up with the measurement for the protein. And so there you need to account for shared peptides. I think Steve mentioned multiple kind of options that you have. And many of the software tools that we use provide all those options. And depending on the study and depending on what you're looking for and the biological question you may have, you may pick one or the other approach to these kind of parameters that you need to set when you're doing your search. There's some additional considerations beyond these basic steps, and one is what do you do with PTM? So how do you identify them? How do you localize where the modification was, and how do you quantify those? And then there are all these various different ways of generating data in the mass spec that all the previous speakers mentioned. So you have DDA, you have DIA, MRM is targeted proteomics. So these are ways of running the mass spectrometer. There are different ways of preparing the sample. So you have label-free, you have metabolic labeling, you have isobaric labeling. So depending on how the samples were prepared and how they were run on the mass spectrometer, the analysis software may need to do different things in order to extract quantification, validation, and identification. So this is basically like a summary of all those things. So this is looking up the result in the back of the book for identification. So you basically search through a database. For validation, determining false discovery rates, I think Steve mentioned you start with the database, you create a reverse database, which we call a decoy, and then you search through everything to figure out the false discovery rate. And then for quantification, you have all these options on how do I combine the information from all my peptides that belong to a protein or a protein group and come up with quantification. So I won't belabor this slide much. So this is the workflow for SpectroMill. I'm not going to go through this, and this is just meant to scare you and show you that it's a pretty complicated process. So processing raw mass spec data, I'm sure processing most raw data, whether it's genomics or other fields, is complicated. You need to kind of understand a lot of things about the data to correctly process it. But I think proteomics data has an even steeper learning curve. There are many kind of nuances which can trip you up, and even like seasoned people can trip and end up with near publications that have incorrect data. So the point here is that you start with your output from the instrument. You have a protein sequence database for the human database or a rat data, depending on the species you're looking at. You extract the spectra, you create your IDs, you validate your IDs, and then you create your combined, the quantified output. And here, there's, I think SpectroMill also includes a lot of other QC metrics that it creates, so you can evaluate how good was your mass spec run, how good were the sample processing, and did you get the best data that you can get. So those can be evaluated by looking at the QC metrics. SpectroMill also has additional tools that will let you annotate, normalize, and format your report so that it makes it much easier to analyze data downstream. So, but the catch with SpectroMill, well, there's one good thing about SpectroMill is that Carl Clauser, who created SpectroMill, is a member of our group. So if we need to change anything or we need to add any new feature, we can go to him and he'll do it in a couple of weeks. The downside is that Carl does not believe in DIA. So this thing only works with DDA. So if people in the group have generated DIA data, then how do they analyze it? So for that, we have created additional software or taken software that other groups have created and made it available for use in the group. So one such piece of software created by Alexei Neshvitsky's group at the University of Michigan is called FragPipe. So this is a collection of a lot of tools that includes database searching for both DDA and DIA, validation using a variety of tools, both for regular peptides and PTM or post-translational modification peptides. You have quantification for DIA, TMT, label-free, and all the other types of mass spec data we mentioned. And it also has some additional downstream visualization and stuff that we don't use in the group. But what we have done is we have taken this entire pipeline and encapsulated it in Terra. If you're not familiar with Terra, it's like a cloud-based platform that is available at the BroadE. I'll provide a little bit more detail on what Terra is when I talk about Panoply, but think of it as like a cloud-based pipeline where you can harness large amounts of large virtual machines in order to analyze lots of data. We also have several other tools for working with DIA. So DIA-NN or DIAN was like a deep neural network based method that was published like a few years ago. That is quite good at analyzing DIA data. Spectronaut is a commercial tool, but because that's primary, the company's primary area of focus, they've put in a lot of effort into creating a tool that has pretty good features and comes up with very good output. So here they have a version that you can run on a Linux computer, and we use that on the cloud. And for this, for example, you can have virtual machines that have 224 CPUs and close to a terabyte of real memory, not disk. And so you can really process large amounts of data with these kind of virtual machines. And then FragPipe, which I mentioned previously, also does DIA data analysis. So any questions so far about raw data analysis? Okay, so now I'm going to switch to Panoply, which many of the other speakers have mentioned. The goal of Panoply is effectively to create a platform where a lot of the state-of-the-art tools are kind of encapsulated in a very easy-to-use manner. And the way we are been using it and the goal with which it was created was that you would create your data freeze. So you would analyze your proteomics data using SpectraMill or FragPipe or whatever. And then you would get your RNA-seq data analyzed using your standard RNA-seq pipeline, genomics data. So this is a proteogenomics data analysis pipeline. And so you get all your genomics, transcriptomics, and proteomics data. You just put all the data into this pipeline and press go. And the idea is that it will run a large number of useful and state-of-the-art analysis approaches, and then come up with a set of reports that will kind of summarize what each of the analyses did, that you can explore interactively, and also provide a lot of like tables and graphs and stuff that explain all the results. And if you need to include something in your paper, you would have a PDF file that you can take and use. So here we also, because this is a proteogenomics file, proteogenomics data analysis pipeline, it takes proteomics and genomics data, and we are currently also working to integrate metabolomics data. So there are raw data pipelines, and actually there is also a link from like the repository of proteomics data to directly get data into the system so that you can run one of these raw data pipelines on the mass spec raw data at some public repository and then seed this pipeline. So it also includes some data preparation modules. So it does sample QC, where you can kind of look at all your samples and calculate basic QC, like correlation between proteome and RNA and stuff like that. And many times we have found that there are sample swaps or sample labeling errors that we can catch here, go back to the data, fix it, or if it was in the lab, we can fix it in the lab, come back again and rerun the whole thing. You can do normalization, filtering, missing value imputation, and several other data preparation tools are included. And then once you have all the data properly prepared, it'll automatically run all these data analysis modules. And we are constantly adding new modules based on what the state of the art is. So we are, like Steve mentioned, part of this clinical proteomic tumor analysis consortium, and many methods are developed there pretty much for every paper that's published. And as the methods mature and we think something is really useful to regularly apply for every cancer data set, we kind of encapsulate it in these modules. And Panoply is designed in such a way that you can add all these modules relatively easily. So I'll go into the details of like a couple of modules in a few slides, but that's kind of the overview. And this was created so that it would be automated. So you put in the data, press go, you'll get the results. but also easy to use. So you should be, it actually provides you a reasonable amount of help to kind of guide you on how to set up the whole thing and run data. So this shows how it's the architected using Terra. So Terra is a cloud-based platform for running various algorithms that's developed at the BroadE. It's based on the Google Cloud platform. And what it does is it creates this thing called a workspace. And in the workspace, you have storage, which they call a bucket. You have all the data and results that you have generated. You have any configurations, parameter settings, and choices for your algorithms. And your algorithms are available as workflows, which you can also put into your workspace. So if you have a workspace and you run an analysis, it's a very reproducible way of encapsulating what you did for your paper or for your research. So you can create a workspace, put your data in the buckets, import workflows for whatever analysis you want to do. And once you do your analysis, the results will also be in your workspace, and you can actually share this workspace. You can share it with your collaborator, and or if you publish a paper, you can make it public so that people can actually come here and see everything in one place. And so we use that model, and here we have created workflows for all the proteogenomic data analysis that I've mentioned as part of Panoply. And the Panoply is a public open source software, so anybody can access it. The code is on GitHub, and there are workspaces in Terra that are publicly accessible that you can clone and start using. So this is, I think I've kind of mentioned this. You assemble your input tables, you clone the Panoply workspace that has all the pipelines, workflows. You upload your data, and then we have a startup notebook that will kind of guide you through the process. And once you do that, everything will be set up, and all you have to do is press go, and it'll run your workflow. So this is like some screenshots of the interface. This is the basic Terra starting page. This is one of the workflows that we have. And this is a screenshot of the start of the notebook, which you will just go step by step, and you'll get your results. The results are available as interactive reports. So they are HTML reports that you can download, and then you can look through them. There'll be a blurb on what was done. There'll be usually figures and tables showing the results, and you can explore that to your heart's content. And like I mentioned, there are a lot of modules, and it creates a lot of output. So it creates quite a large zip file that has all your inputs, but it also separates out the reports so that you can quickly look at the reports and then dive into any of the independent modules that you want to look at in more detail. And this has been applied in a lot of cancer publications that are part of the CPTAC. And for CPTAC 4, I think one of the requirements was that we have to apply it to every study. So it's quite extensively used. And members in the group use pieces of it for their analysis, even if it is not cancer related or CPTAC related. So I'm going to go through like a couple of modules pretty quickly to give you a flavor for what are the kind of things that are in Panoply. But I'll focus on things that are of more general use rather than just specific to cancer or very focused application. So Steve and others mentioned that when you generate proteomics data, especially post-translational modification data like phosphoproteomics and so forth, you get a lot of missing values. Because if you found a phosphopeptide with a specific phosphorylation site in one of your samples, how likely is it that you're going to find the same thing in the other 100 samples that you have? So some of the peptides you do find, but many times there is a significant fraction of missing values. And so the question is, what do you do with missing values? Do you throw away the data, or can you somehow salvage it? So what we do in our group is we filter phosphopeptides or proteomes to make sure that there are a reasonable number of observed values. So we usually say at least 30% of our samples should have some observation. So 70% could be missing. And then depending on the type of algorithms that we want to apply and the type of analysis that we want to do, we impute missing values. So one of the modules in Panoply is a missing value imputation module. And we have done quite a bit of work on trying to figure out what is the best way to impute this kind of proteomics, especially post-translational modification data. And so the best methods that we have come up with are implemented. And this is just a summary of the various methods we tried. I won't go into the details, but there is a algorithm called MICE, which stands for multiple imputation by chained equations. And that works really well for imputing both global proteomics and post-translational modification data. And that's the method that's currently implemented if you need to impute data. You don't have to impute, but if you do, we have a good method. Some of the analyses can be done without imputing, but for some, it might help to impute. The other kind of method in Panoply that's been extensively used and kind of applied to almost all cancer studies is this thing called NMF multi-omic clustering. So NMF stands for non-negative matrix factorization. If you're not familiar with it, matrix factorization is a way of taking your data, which is this large square on the left, and breaking it into two matrices. So you can think of this as a big matrix. You take this and you split it into two matrices. The way it's split is that this left matrix, which we call the amplitude matrix, kind of is a clustering of the molecules of interest. So it's a clustering of your genes or proteins or phosphosites or whatever. And the other matrix called the pattern matrix is a clustering of your samples. So if you just look at the pattern matrix, you will get a clustering of your samples based on this matrix factorization method. And so we call that NMF clustering. And the advantage of NMF clustering is that it's been shown to be pretty robust and it works well on a large number of data types. So what we do is we take all our data like copy number, gene expression, proteome, phosphoproteome, ubiquitylome, we stick all of it together and do multi-omic NMF clustering to come up with clusters that hopefully capture information in all the omes, not just the transcriptome or not just the proteome. And there have been examples where we have come up with subtypes that have not been observed just using RNA-seq data and have survival implications in terms of one of the clusters. So here you can see the dark blue is luminal A type breast cancer. So the luminal A type breast cancer is split into two groups. And in one of the groups, it has a much worse survival compared to the other group. And this was not previously known or observed just using the RNA-based typing, which is captured in the PAM50 for breast cancer. We have observed similar things with many other cancer types, but this is an example I thought that was very unique. We also have so many of you have probably heard of gene set enrichment, where if you have done like a comparison and you want to see which pathways are enriched in your comparison, you do gene set enrichment analysis. The problem with gene set enrichment analysis is that you have to have your data at the level of genes. So your data must have each row in your data must be a specific gene. So now we are measuring proteins. So first off, mapping proteins to genes is going to lose some information because at the protein level, you get a lot of isoforms. So all those are lost when you map to genes. At the phosphoprotein level, you lose even more information because at the phosphoprotein level, the mass spec gives you phosphosites. So you have individual sites. And then a set of sites map to a protein isoform. And then a protein isoform maps to a gene. But if you go in reverse, a gene will map to like many isoforms and even more phosphosites. So you're doing so much work to get all this PTM phosphosite information, and you're throwing it all away to do gene set enrichment analysis. So what we did in our group was we created this thing called post-translational modification site-specific enrichment analysis. We call it PTM SEA. And here what we do is we work at the phosphosite level, and we have created a database that we call PTM-SIGDB, which is a database of pathways at the phosphosite level. And an additional thing that we do compared to the traditional GSEA pathway databases is that we have the directionality of the change also included in the database. So you do a scoring based on things that are going up, you do a scoring based on things that are going down, and then you combine the two scores to come up with the final enrichment score for PTM SEA. So we have found that this does provide quite a bit of additional pathway level information, and is definitely using more information in the phosphor domain. The final tool I'm going to cover is this thing called Scion, which Natalie created, but I'm presenting because it's part of the Panoply assembly rather than some other assembly. But here, what we do is it's a multi-omic network analysis algorithm. So we start with two different ohms that are given as input. So it could be like the proteome and like gene expression data. You could restrict it to like just transcription factors or any other subsets that you want. So you start with those two, you put the two matrices together and you cluster them. And you get some number of clusters, and for each cluster, you infer a network for that cluster. And then you try to combine all those using cluster specific, so using hub genes. So genes that are hubs, you're trying to look across all the clusters and try to combine them. And then ultimately you use some sort of a permutation test to make sure that you keep only those edges that are really different from random, right? So many times if you have so much data and you do so much manipulation of the data, you can end up with links and associations that are not really true, that could have happened just by random chance. So you try to filter those out, and then you come up with a much more manageable hairball, I guess. So, so it's a difference between like hairballs from 4 cats versus just 1 cat or even less. And the point is you will get more information out of it, and you can dissect it and look at it more easily, and hopefully get some level of like biological insight. So this is a result for a lung adenocarcinoma data set that is a joint effort between CPTAC and a group in Taiwan. So we had like about 400 tumor samples, and this is like a clustering for that. And we have looked at the clusters, and there are a couple of subclusters that seem interesting, and we are kind of trying to explore that. So that is Panoply and some of the associated tools in Panoply. So any questions so far? Otherwise, we'll switch to Natalie. Yeah.
How do you deal with the variance? Because once you use the max algorithm, right, but you're combining, so it's sort of making the assumption it's a single set, but in essence, it's actually multiple.
For the multiple missing values?
How do you deal with the variance around that?
We ignore the variance. So we just take the mean of, so we impute like 15 times, let's say. That's the multiple imputation part. And then we just take the mean of the 15 values. The variance is ignored. So let me actually preface it properly. So there are mathematical methods. So if you do regression, or if you do like specific types of linear models, you can use all the 15 and systematically take into account the variance that you have seen between the 15. But if you want to do clustering, you can't really do variance, account for variance in clustering. So I think one of the things I showed was the output from this multiple imputation method is a single imputed data set based on the averages that you can use for things like PCA and clustering and algorithms that cannot deal with the variance of the missing values. But it also gives you the 15 data sets with the multiply imputed values. And for linear regression and other kinds of modeling techniques where you can actually take that into account, we do take that into account.
But then how do you test the how do you do sensitivity analysis for that? Because you're making the assumption it's missing at random, right?
So yes, so some of the proteomics data is actually missing not at random because it's based on intensity. So, but one thing we have done is, so I kind of glossed over that. In evaluating these missing value techniques, we have simulated data that is from missing not at random mechanisms, missing at random mechanisms, and we have shown that this method works reasonably well under both situations. And so we just apply this one method. Otherwise, it's much more trickier. So if you can assume that your data is only missing not at random and is driven by low intensity, there are some other methods, like so MaxQuant, for example, uses a method where they look at all the low, the distribution of proteins with low expression, and then they build like a Gaussian distribution of that, let's say with some mean and variance, and then they shift the mean by like 2x less or something, right? So they shift it towards the lower values, and then they generate a set of random values with that distribution to fill in the missing values. But there you are explicitly saying that all my missing values are based on low abundance. We have found that that's not the case 100% of the time. A majority of them are, but they can still be generated by other factors like stochasticity and so forth. So we have found that using something like mice accounts for both in a reasonable way. Any other questions? Okay.
Okay, I think that works. Can everyone hear me? Okay, cool, awesome. Okay, so now I'm gonna sort of transition us from what Mani talked about to things that are, I think, a bit more applicable for sort of these more smaller, more customized experiments. Maybe you don't have hundreds of tumor samples. Maybe you just have like one experiment where you have 3 replicates of like a wild type, 3 replicates of a knockout, and what can you sort of do with that? So the first thing I'm gonna cover is this tool we have in the lab called Prodigy, which is actually our Shiny app. So this app can basically be downloaded from the GitHub repository, which is down here, and run locally. And it basically allows you to do many of the sort of initial QC things you would do with your data. So like looking at distributions of your data, doing things like log transforming your data, normalizing your data, doing statistical analysis and basic visualizations. We also have this hosted on a Posit Connect server, formerly our Studio Connect server. And that basically allows us within our group to host this on the web so that members of our group, as well as collaborators, can use this tool to basically import their data, do the analysis, save the session for later, and then they can actually go back to the website, load their session, and continue from there. So they don't have to constantly sort of do the analysis, save the data, and then maybe they decided they wanted to try another normalization or something like that. So what can you do in Prodigy? As I mentioned, you can do QC. So you can basically look at, here are my data as they are, and what happens if I try to do a median and mad normalization or some other normalization to adjust for things like differences in the mean or variance of my expression. You can also do things like filtering. So as has been mentioned many times, in proteomics and especially PTMs, we have lots of missing values. And so incorporating those features that have lots of missing values could impact your analysis. So you can play around with sort of, you know, if I use a more stringent or less stringent cutoff, what features am I losing? How does that impact my analysis? We have some sort of simplistic statistics in here. So 1 sample t-test, 2 sample t-test, as well as f-test for when you have more than 2 groups. And then a lot of visualization. So for example, here's a volcano plot from a 2 sample t-test. They're interactive. So for example, if you want to know what's this protein way out here, you can actually click on it and it will tell you, you know, the gene symbol that that protein is associated with, as well as the p-value and the fold change, that kind of stuff. And then also PCA plots. So this is really a nice way to visualize if you have replicates in groups, you know, are my replicates or clustering together? Are my groups very different from each other? These sort of basic visualizations. So what is the workflow of going from data and actually getting all these plots? So Prodigy works with a type of file that's called a GCT file, which stands for gene cluster text file. And so what this file is it's a file that not only contains your data, but it also contains your metadata, all within the same file. So you'll have metadata for your features, such as the protein isoform, the gene symbol it maps to, things like that, and then features for your columns, which would be, is this a wild type or a knockout? What batch was this processed in? And by having it all together in one file, what Prodigy can do is it can basically pull apart the data from your metadata. And so a lot of the colors that you saw
on this slide are because in the GCT file, it's already known what sample, for example, is associated with each of these groups. So all that kind of color coding can be done automatically rather than you having to specify what sample, which color. So it saves a lot of time. And then there is some built-in gene mapping for some organisms. Of course, you can use this on any organism. If you import a column that has a gene symbol, Prodigy will make sure to try to map your genes appropriately. And then you kind of have choices once you sort of look at data normalization and filtering. If you want to, you can go through statistics and do some exploratory data analysis. But maybe you just want to normalize your data and then you want to go do a linear model with it. That's also possible. You can totally skip this middle part, go straight to checking your QC, and then just go straight to exporting your data. And for exporting your data, there's lots of options. You can export another GCT file. So that would be just like the original GCT file that has, for example, your normalized data in it. Or if you did statistics, it might have your p-values in it so that you can go do GSCA or something like that. You can also get an Excel file because not everybody likes to use GCT files. So if you just want your data without all the problems of a GCT file, that's an option too. And then for visualizations, all the visualizations gets exported as high-res PDFs that come in a zip file. So if you see a visualization, you're like, oh, I really want to put that in my figure, in my paper, you can download the zip file, get the PDF, pop it into Illustrator, and boom, figure's done. I'm going to quickly go through these slides because there's a lot of information on them. The main point is just to show you these are all of the things you can do in Prodigy. So it really is meant to be a one-stop shop that covers a lot of different applications in proteomics. So as Steve's mentioned, a lot of times we work with ratios, but sometimes, especially with DIA, we're working with intensities. So we have that built into Prodigy where you can just basically flip which one you're working with. Options for log transformation that are pretty common, lots of options for normalization because there's not usually a one size fits all. And then as I mentioned, a couple of the filtering options. And then in terms of tests, as I mentioned, we have a 1 sample, 2 sample, and a moderated F test. You can choose how to filter. So you can choose if you want to use an adjusted p-value or a nominal p-value. You can play around with the cutoff so you can see if you're more stringent or less stringent, what proteins get kept or get knocked out. And as Mani mentioned, at the end of the day, all of these are really just linear models that have been implemented using the lemma package. So if you've used lemma before for RNA-seq or something like that, it's the same sort of statistical framework, just applied to proteomics data. And a lot of the sort of methodology that's in Prodigy could be used in other omics data. Like you could potentially use Prodigy on like RNA-seq data or something like that. And then all of the results sort of get put into tabs here. So if you're really only interested in like, I just want to load in my data and get a PCA plot, you can just go straight to the PCA plot, look at the plot, format it how you want, and export it and go. So you can spend as much or as little time in the application as you need. So we're always thinking about how we can improve these methods. And so one sort of limitation right now of Prodigy is that you can only load 1 file at a time. And if you remember back, we were talking about this Monty protocol we have, which allows you to get global proteome as well as all of your PTMs. So right now, if you run Monty and you want to go look at your data in Prodigy, you have to do a separate Prodigy session for every single one of your ohms, which is kind of annoying. So we're working on expanding Prodigy so that you have a multi-omics capability so you can put all of your GCT files in at the same time, analyze them all together, and then export everything together so you don't have to do it multiple times. We're also working on adding some additional modules that we use in the lab. So one of these is our linear model app, which I'm going to get to in the next section. We also have this multi-omics heat map viewer, which was created by Stephanie Fartani, who was a previous associate computational biologist. And basically this allows you to visualize multi-omics data in a heat map. So you can see for a particular protein, if you detected it at the RNA, the protein, and the phosphosite level, how those different features are changing over your sample. So you kind of get a nice visualization, as well as other quality of life changes. So Prodigy is something that's been in the group for a while, but it's constantly growing, constantly improving. And like I said, it is available on GitHub. So if this is something you feel would be very useful for you, even if you're not working in proteomics, maybe you're working in RNA, it's something that you could go to GitHub, you could download today, and you could get started using immediately. So any questions on Prodigy? If not, I'll move on to the last section, which is our sort of a computational consulting. So these are sort of more customized analyses that we do for the group. And so I'm going to start with linear models. So I would say after things like Panoply and Prodigy, probably the next most common thing we do is to design and implement these linear models for a lot of these smaller, more customized studies. And the models that we sort of build in the group can range from very simple to highly complex. So as I mentioned, for some of the most simplest linear models, some of them you can just go to Prodigy and do in Prodigy. Then there's sort of this intermediate group where maybe you have something like a 2-sample t-test, but you have some sort of controls. That could be modeled as a linear model with and without interaction term. Or perhaps you have something like a 2-sample t-test, but you have replicates. And so you want to treat those replicates as a random effect in the mixed effect model. For these, we have developed another R-Shiny app that sort of covers these intermediate cases. As I mentioned, one of our ongoing projects is to sort of integrate this with Prodigy. So hopefully in the future, these sort of two top categories can be combined together and used together. And then finally, sometimes there are just some experimental designs that we can't really cover with sort of these standardized approaches. So things like a thing that we run into a lot of times with proteomics, especially in these large dimensional projects like these CBTAC projects, is batch correction. So a lot of times these samples are run in different batches. Sometimes because of life events like COVID, those batches could be separated by months or even years. And then when you go to analyze these data, you suddenly have a batch effect where your samples are differing just because there was a large amount of time between them. And so that's sort of something that we have to address a lot in our group, just given the intricacies of all these projects. And then sometimes there are just models that maybe you have lots of different covariates, maybe you have different interaction terms, and those are lots of times where we will move into just writing the model out and then analyzing the model in R. So we also have the capability for these more complex experiments to do something more custom. But in the cases that are more standard, we have really worked on putting together these reproducible and easy to use applications so that you don't necessarily have to come to us. You can use the application to get the results that you need. Another thing that is kind of common problem in our group is that you can imagine that with these large consortium projects, so like Mani mentioned, there's one that has like, over 400 samples, and in our group, we have sort of over 200 samples. You have to start thinking about, they're gonna have to be run in different batches. How do I randomize those batches so that I reduce any concordance between things like age and sex and covariates in my data and any batch effects? And so a lot of times in the past, this has actually just been done by hand. So people will make this giant Excel sheet and they'll color everything a different color and it looks like a unicorn threw up. And then they just kind of work out something that looks nice and they say, okay, that's randomized enough. But we kept thinking, as our data sets gets larger, as these batch effects become more common, is there a way that we could somewhat standardize this? So C. Williams, who's another associate computational biologist in the group, came up with this idea to sort of automate this randomization. And they came up with this app, which is called ShinyStrat. So ShinyStrat is another R-Shiny app. The input to the app is actually just an annotation file. So you basically have for each of your samples, all of the covariate information. So like age, sex, who did the lysis, things that you want to control for. You can select what exact annotations you want to randomize, and you can also give it the size of your batch, and it can be any size. So like in proteomics, for example, if we're doing a TMT experiment, we might have 16 or 18 samples. They have other groups have used this for like when they have a 96-well plate, and it also works really well. And then what happens in the background is that the annotations are randomized and they're assigned to sort of the best randomized batches using a normalized chi-square metric. And then the output is not only a table of here are your samples and here's how you should randomize them in each batch, but also a report that basically shows you how good was your randomization. Are there any samples that ended up being unbalanced? And can you sort of go back and fix that? And so this has been routinely used in our group for TMT plex design. There's also one of the consortiums that we work with that Shanka mentioned earlier, the MotorPack Consortium on exercise, and they have actually implemented this pool-wide for their consortium. So now it's currently being used by the entire consortium across all different omics types to randomize their samples, so that hopefully we can sort of keep these batch effects to a minimum and have our randomization approaches standardized. So no more rainbow Excel files, I'm sorry to say. But the reports are very rainbow and pretty, so there are still rainbows. And I'm gonna wrap up with sort of, again, looking more forward to, you've heard about a lot of new technology that's happening in the proteomics field. And with new technology also comes new computational methods. So we're constantly sort of innovating in our group to sort of come up with new methodologies to support these technological innovations. And so the first two, I don't think we touched quite a bit on today. I think Steve mentioned them briefly into introduction, but Olink and Seer are these other platforms. So for Olink, Mani has actually been doing a lot of work on this Olink proximity extension assay panel. For Seer, we have these multiple nanoparticles that I think Steve highlighted in the talk. And so one thing we have to think about is, how do we combine the protein quantifications when we have, for example, 5 different nanoparticles? So that's something we've been working on. PISA, which you heard a lot from Namrata. So now with PISA, we have a completely different output, right? Now we have sort of these dose-dependent response curves. And so fitting those curves and coming up with what's significant in this space is a lot different than, for example, when you have just protein quantification and do like a t-test. Single-cell proteomics, as Claudia mentioned to you. So one of the things that we've done is there's been a lot of work in the single-cell RNA-seq field regarding things like normalization, because now that we're working at the single-cell level, a lot of the assumptions and methods we use for bulk level data just don't really apply to single-cell. So we've been experimenting a lot with, can we potentially take previously existing normalization approaches from the single-cell RNA field and directly apply those to single-cell proteomics. And then spatial proteomics, which is something that Claudia touched on, is something that we're also, kind of working on. So as these projects sort of develop all these new modalities, we're constantly innovating as well. It's just a cycle. So with that, I'll end and here you go. Here's the famous photo. Again, it is now burned in your brain forever, I'm pretty sure. So again, want to thank the entire computational proteomics team. So I think we've mentioned most of our current members as well as our alumni. And then we also want to thank people in the group who maybe aren't officially members of the computational team, but they do a lot of computational work for us. So a lot of times there are people outside the computational group who have a great idea and they want to implement it, and we say, go right ahead. So again, just really highlighting how this is a collaborative environment.
And I especially thank you for attending this and for your questions. And don't hesitate to get in touch with us if you're interested in anything that you heard. We'd love to talk to you. So have a wonderful weekend. Thanks again.